Welcome to this tutorial on the thalamus. Now that we are into our studies of our sensory systems and we will progress from there to our studies of the motor system, we're going to encounter an interesting structure that's deep near the center of the brain called the thalamus. So I think it's worthwhile for us to think in general terms about the structure and function of this part of the brain. So thank you for joining me for this discussion. This pertains to several of our core concepts in the field of neuroscience. Again, the complexity of the brain confronts us every time we consider one of its component structures in relation to others. We will be talking about the derivation of the thalamus in embryological terms, which reminds us that the circuits in the brain that are foundational for function are all encoded in the information that's present in our genome. Now, those circuits are formed and shaped and changed in very interesting ways through activity-dependent mechanisms of plasticity, but the basic architecture seems to be fundamentally genetically determined. And as a consequence of this genetic endowment and the accrual of our life experiences and how all of these factors self-organize in the developing brain, we have this fantastic capacity to ask questions, such as what has led you to take this course with me? You've been endowed with a curiosity that arises from the structure and function of your own networks within your brains. All right, well, our objectives for this particular tutorial are that I want you to be able to briefly discuss the embryological origin of the thalamus, and I want you to be able to identify its location in the human brain. And lastly, I want you to be able to have just a very short discussion with a friend or a family member regarding the functions of the thalamus in the mammalian brain. Okay, concerning its embryological origins. The thalamus is derived from that forward part of the developing embryo that will form what we call the forebrain. And in embryological terms, the forebrain is the prosencephalon. Let me remind you that the nervous system develops from the walls of a tube. And those walls have an anterior end. And here is the anterior end of the developing neural tube shown schematically. And in a more realistic view of the embryo, there's the anterior end. The posterior end is the inferior aspect of the spinal cord. But on the anterior end, we have a large swelling that begins to form. And as embryology proceeds, that swelling subdivides. There is a very large telencephalic vesicle that forms, and here is where we will derive our cerebral cortex, or our basal ganglia, much of what we consider when we think about the mammalian forebrain. But the more posterior part of this structure is what forms the thalamus. This is called the diencephalon. And there are lots of other interesting developments that emerge out of the diencephalon. One of them is shown here, and that's the optic vesicle. Here it is in schematic form. The optic vesicle goes out to form the neural elements of the eye, the retina. So, believe it or not, the retinas in our eyes are truly brain. They are derived from the diencephalon, a division of the posterior forebrain. So, in this view, here is our diencephalic region near the posterior part of the forebrain, just between the telencephalon and that middle brain, the mesencephalon. And as we see the consequence of this brain development, we find that the diencephalon is found very much near the center midline of the brain. And in the more posterior and dorsal aspects of the diencephalon, we have the region that we call the thalamus. More ventral and anterior is the part that we call the hypothalamus. There are other divisions that are of less importance to us, at least right now in the course. We'll speak about them as needed as we progress. But for now, I want to focus in on this division of the diencephalon that we call the thalamus. And again, just to remind you of where this is from, Embryologically speaking, the diencephalon is derived from the prosencephalon. It's the posterior part of that large anterior swelling that sits in the transitional region between the telencephalon and the middle brain, or the mesencephalon. And then the diencephalon goes on to subdivide into this dorsal thalamus and a more ventral anterior hypothalamus, both of which form the lateral walls of the third ventricle. Okay, so that's where we are from an embryological perspective. What about from a neuroanatomical point of view, thinking about the whole human brain? Well, let me now show you in a phantom view of an adult human brain what we saw in an earlier illustration of a developing brain, and that is that the thalamus ends up very near the center of the forebrain. So here we're looking at a phantom view from the lateral aspect. This orange curved structure is part of the basal ganglia, and what we see is that the thalamus is tucked in on the medial side of the basal ganglia, and it extends just a bit posterior to it. So if we were to look at some cross sections, what we would discover is that the thalamus sits as something of an egg-shaped structure near the midline, but also very near the center of the forebrain. And just to the lateral aspects of the thalamus is a big bundle of white matter known as the internal capsule. And on the other side of that internal capsule are components of the basal ganglia, specifically two divisions out here called the globus pallidus and the putamen. Just above the thalamus, we find the lateral ventricle, this large space that sits just above the thalamus, but also just above the thalamus and to its lateral margin is an additional component of the basal ganglia called the caudate nucleus. Well, let's look inside the thalamus if we can, and I'll point you to this illustration which indicates that the thalamus is really not one thing, it is dozens of things. It is dozens of discrete collections of cells that each maintain their own set of input and output relations to other parts of the nervous system. So the thalamus comprises some 20 or 30, depending upon your tolerance for dividing up different parts of this structure. These 20 or 30 individual nuclei represent discrete pathways that are providing inputs to the thalamus, and then the thalamus in each of these subdivisions gives rise to outputs that are directed mainly to the cerebral cortex. Well, how can we make sense of these 20 or 30 subdivisions of the thalamus? I would suggest a pretty simple anatomical framework for doing so. When we look at the human thalamus, we see that it's roughly divided by a white matter layer that runs right through the middle of it. It sort of has a Y shape, if you can appreciate this as a shape resembling the letter Y. There's an anterior part of this thalamus in the crook of this Y that contains a group of nuclei that we call the anterior nucleus, or the anterior nuclear group. This part of the thalamus maintains interconnections with the front part of the frontal lobe, a region that we call the prefrontal cortex. Specifically to a component of the prefrontal cortex in the cingulate gyrus. The medial part of the thalamus is comprised of a set of nuclei that project to other parts of the frontal lobe and the insular cortex and even the anterior part of the temporal lobe. These are all components of that prefrontal cortical network that are more broadly distributed. There's a significant nucleus in this group called the medial dorsal nucleus of the thalamus that helps to define what we mean by prefrontal cortex. Now, on the lateral aspect of the thalamus, we find most other cell groups that are going to be of interest to us in this course. These include a variety of nuclei that project to the somatic sensory regions of the parietal cortex, the visual regions of the occipital cortex, the auditory structures, in the temporal cortex, and even the motor structures in the posterior part of the frontal lobe. So it's in this lateral group that we find many of our sensory pathways that we'll be talking about over the next several tutorials, running through the thalamus, relaying information to the cerebral cortex in an areal specific pattern. Now, there's one other set of nuclei that I'd like to highlight for you, and these are a collection of small nuclei that are actually buried within that white matter lamina that forms this Y shape. These nuclei, in addition to some nuclei that are around the midline of the thalamus, tend to project very broadly to widespread regions of the cerebral cortex. We don't think of them as conveying specific signals, such as our sensory and motor nuclei that we find out here in our lateral group. Rather, we think of these midline nuclei and interlaminar nuclei as being primarily modulatory in function, rather than conveying specific information. Now, let's use this anatomical framework to consider the function of these different kinds of nuclei that we've just described in the thalamus. The most widely known set of nuclei in the thalamus are those that receive incoming or ascending sensory signals, and the function of these nuclei is fundamentally to relay that information to the respective divisions of the cerebral cortex that are defined by the input patterns of those nuclei. So, for example, there is feed-forward transmission of ascending sensory information through nuclei of the thalamus that are concerned with somatic sensation. They receive inputs from somatic sensory pathways that are arising from the spinal cord and from the brainstem, and they send their signals into the somatic sensory division of the cortex, namely the postcentral gyrus. The same principles are seen in the organization of the pathways for vision, for audition, and for our sense of equilibrium, our vestibular sense. Each of these senses have relays through specific nuclei of the thalamus. In fact, those divisions of the cerebral cortex that are dedicated to processing inputs about a particular sensory modality, such as the somatic sensory cortex, the visual cortex, they are considered primary because they're the first cortical area to receive input in this ascending sensory stream. And the reason they're the first is that they are the recipients of a specific set of thalamic inputs that arborize in a particular sector of the cerebral cortex. So, in this more schematic view of thalamic function, what I'm talking about are what we might call first-order thalamic nuclei, or sometimes they're called specific thalamic nuclei. These are nuclei that are receiving sensory or motor signals and sending inputs up to cortical networks through very specific and very robust connections that terminate in the middle layers of the cerebral cortex. These first-order thalamic nuclei are typically those that we find in the lateral division of the thalamus. Well, in addition to these first-order nuclei, we can also recognize higher-order thalamic nuclei. These are thalamic nuclei that receive primary input not from ascending sensory or motor pathways, but from descending pathways that arise from the cerebral cortex itself. So these are thalamic nuclei that seem to participate in cortico-thalamo-cortical loops. That is, the distribution of signals from one region of the cortex to another through a connection in the thalamus. And lastly, I would highlight those set of thalamic nuclei that are typically on the more medial part of the thalamus, but also those nuclei buried within that lamina of white matter, also in this category, that we might consider to be modulators of cortical function. These are what we call nonspecific thalamic nuclei that project not to the main input layer of cortex layer 4, but more broadly, typically to the upper layers of the cerebral cortex. And these more diffuse projections seem to be very important in modulating the state of the cortical network. So rather than providing specific sensory and motor signals or distributing particular higher order processing signals, these more diffuse axonal systems emanating from the midline and intralaminar nuclei of the thalamus perhaps have a role to play in modulating cognitive functions such as attention, or perhaps modulating our level of arousal. So these are not specific nuggets of information that are being transmitted along thalamocortical lines, but rather it's a modulatory signal that can have a widespread influence over large-scale cortical networks. Welcome back, and welcome to this tutorial in which I hope to provide you with an overview of the cerebral cortex and cortical circuits. I know I've said this repeatedly so far in the course, but we truly are dealing with the most complex organ to be found in the human body. And when we talk about the organization of the cerebral cortex and cortical circuits, we're largely looking at the product of genetic instructions that are determining how these circuits are going to form and how they provide for the foundation of all the amazing functions that our cortical networks perform for us. One of them is we are endowed with curiosity, with a sense of wonderment, and I hope that capacity is being well exercised so far in this course. As we turn our consideration to the cerebral cortex and cortical networks, we can't help but to be reminded of the importance of this part of the human nervous system for health and wellness and optimal functioning. And we can't help but to be reminded of the hundreds of thousands of people that are living worldwide with neurological disability because of some problem in the development or in the ongoing health status of the cerebral cortex and its networks. With a reminder of those core concepts in the field of neuroscience that are at stake today, let me point you to our learning objectives for this session. I want you to be able to discuss the embryological origin of the cerebral cortex. I want you to be able to discuss differences in the cellular architecture that we find from one region of the cortex to another. And I want you to be able to discuss not only what's different from one region to another, but what's conserved. And that would be the anatomical organization of what we'll call the cortical microcircuit. And lastly, I want to give you a bit of an acronym that might help you remember what are some of the principal functions of that microcircuit. And I call that the ACC of the cortical microcircuit. Okay, well, let's begin with just a bit of embryology. Now, the cerebral cortex and all the networks contained therein arise from the most anterior swelling that develops in early embryonic life. And that anterior swelling of the developing neural tube is called the prosencephalon. I'll just remind you that the nervous system is derived from the walls of a tube. And the anterior end of that tube is closed, and that's what we call the prosencephalon, or the forebrain. So it's going to be a massive swelling that will form at the anterior end of this developing neural tube. As we proceed on with gestation, this prosencephalon further subdivides into a diencephalon and a telencephalon. And it's from this telencephalic vesicle that the cerebral cortex will develop. The cerebral cortex basically becomes this outer rind of tissue, this outer wall of this developing telencephalon. As we progress in human gestation into the third trimester, the outer wall begins to take the shape that we are more familiar with from our journeys into the adult human brain. So what we see now is a cerebral hemisphere with some of the primary sulci or fissures beginning to take shape. So in the third trimester of pregnancy is when we begin to appreciate the central sulcus, the lateral fissure, and many of our other primary sulci and gyri that we saw when we looked at the human brain together in the lab. And as we look inside this brain, if we were to take a cross-section, what we would observe is an outer cortex, which means bark, like the bark on a tree, the outer rind of tissue. So the cerebral cortex is this outermost layer that we observe when we look through a cross-section through the forebrain. Well, it's really incredible to consider that the closed anterior end of the neural tube eventually takes this marvelous shape of the cerebral cortex in the human brain. So it really is one of the grand achievements, both in the evolution of the mammalian nervous system, but also in the development of each of you. And we should just never forget the just incredible achievement in mammalian biology that's represented by the human cerebral cortex. Well, part of the story of how that cortex came to be has to do with many cycles of cell division in early nervous system development. And so there is, as you might imagine, an exquisite set of control mechanisms that are responsible for producing the neurons that are needed to populate the nervous system. And for reasons that are not fully understood, it seems as though the strategy for creating the nervous system, and largely that cerebral cortex, is to create about twice as many neurons that will actually survive the process of brain development into early childhood. And believe it or not, you've had about twice as many neurons in your brain during early embryonic life than what you now have. And that's true of those of you that are younger, and true of those of you that may be older, certainly. So there is a process in brain development that we'll talk about in due course, whereby cells are programmed to die. And some of that cell death seems to be the result of the failure of some competitive mechanism. Some of it occurs for reasons that no one quite understands. But be that as it may, the proliferation of cells is obviously a key component in building a brain. So this happens in a region of the central nervous system that's very close to the ventricles, very close to that lumen of the developing neural tube. There's an interesting dance that we'll come to talk about in a little while as the cell cycle proceeds, where nuclei divide and DNA is replicated in different zones within the wall of that neural tube. But eventually, cells exit the cell cycle and become what we call neuroblasts. These neuroblasts go on to differentiate into neurons and glia. Meanwhile, a progenitor cell is left behind that can continue to divide or perhaps differentiate eventually into a stem cell that may remain competent to re-enter the cell cycle at some later point across the lifespan. Well, after many cycles of mitotic activity, the neuroblasts that result will migrate over some vast territory within the central nervous system. And for the building up of a cerebral cortex, that is an outer bark, there must be a migration of cells from this inner region, close to the ventricle, to this outer bark, where a cortex is going to be built. And we call that developing cortex the cortical plate. So here, very close to the ventricle, is the site where the cells divide and the neuroblasts first exit the cell cycle. Well, in order for these neuroblasts to get from the ventricular zone to the presumptive cortex, that is the cortical plate, they migrate. And they migrate along a scaffold provided by radioglial cells. Radioglial cells extend a process from the peel surface down to the ventricular region of the wall of the neural tube, and it provides a means by which a neuroblast then can migrate by shimmying along this fiber. So through this migratory stream, the neurons that are in the process of differentiating can come to populate this cortical plate. And as a consequence of this grand migration of neuroblasts from deep within the walls of the tube to this outer bark, this outer cortical plate, we end up developing a sheet of cells that reside out here in this outer region of the developing wall of the neural tube. Now, this sheet is actually continuous across the entire cerebral mantle. You might not appreciate that if all we had in view was the adult form of the human brain, but everywhere you see cerebral cortex, we have a continuous sheet of cells. And as I've told you elsewhere, if we take the amount of cortex that can be found in one cerebral hemisphere and flatten it all out, we'd have something about the dimensions of what in most countries is a medium-sized pizza dough, or a medium-sized pizza. Now, the dough of that pizza would have to be very thin. Here in the United States, we call that a New York-style pizza with a very thin crust, because after all, the cerebral mantle, the cerebral gray matter, is only a few millimeters thick. In some parts of the world, the pizza is made in deep dish style, so that wouldn't be a good model for the cortex of the hemisphere. We'd have to travel to Manhattan and enjoy that crispy thin crust pizza that's characteristic of that part of the world. Well, anyway, I hope you can resonate with this picture of a single sheet of cells that is folded into the shape of the four lobes of the cerebral hemisphere. So I want to emphasize for the purposes of this tutorial that this sheet of cells, it's not just one cell thick, of course. There are actually many hundreds, perhaps even thousands of neurons that constitute the thickness of that sheet. And we can appreciate that when we look under the microscope at the cerebral cortex in its adult form that has been stained in various ways to demonstrate the presence of neurons. So I'd like to show you some images that I prepared myself in my own lab. And on the left, what we have is a preparation that shows us the presence of what's called Nissl substance within the cell bodies of the neurons and the glial cells that populate the cerebral cortex. And on the right, we have a stain for a neurofilament that is especially enriched in large neurons. There are a variety of ways that we could prepare stains of the cerebral cortex that highlight its features, and they all show a similar theme, and that is an abundance of neurons that populate that cortical sheet, but those neurons are ordered and they're structured into layers. Now, sometimes those layers are not so easy to appreciate, but nevertheless, histologists over the years have agreed that it is possible to recognize typically 6 layers in most divisions of the cerebral cortex. The first layer is considered just this outer layer, which is right up against the peel surface. So this is the peel layer out here, and just below the peel layer is a region where we don't find a whole lot of neurons. What we find here is actually a whole bunch of axons and myelinated axons. So we can think of this as a little bit of white matter at the very extreme edge of our cerebral cortex. Well, just below that layer 1, where we don't have many cells, we tend to have a pretty distinct population of neurons that have a particular shape and form that are not particularly obvious to us here in this Nissl stain, but through other staining methods we might see them stand out just a little bit better. So that's what we call cortical layer 2. Cortical layer 3 is actually a broad layer that extends for some distance here, and people have subdivided this layer in various ways in different parts of the brain. We won't worry about that. Here I'll just highlight that this is one place where we find these larger pyramidal neurons that have enriched concentrations of this neurofilament protein that stands out here to the right. But in stark contrast, we come to the next layer of cortex, which is called layer 4. Now, layer 4 is really important, and I hope that you can recognize its importance as we talk through our sensory systems. Layer 4 is the target of our first order or our principal thalamic nuclei that are sending signals into this cortical network. So each of our sensory systems, for example, will have a region of the thalamus that sends its inputs into layer 4. So layer 4 is what we call the thalamic recipient zone. It's the first place that gets information from the thalamus that then cortical networks can operate upon. And as we see in the Nissl stain to the left, layer 4 is populated by smaller cells. These cells are often called stellate cells because of their star-like appearance, and sometimes they can be so dense that they sort of look like grains of sand or grains of salt in a salt shaker. So we sometimes call layer 4 the granular layer of the cortex because of the appearance of these grains of sand. Now notice that layer 4 is almost completely devoid of large neurons expressing this neurofilament. So layer 4 actually stands out perhaps a little bit better in the image to the right than the image to the left. Well, right below layer 4 is of course layer 5, and layer 5 is home to some of the largest pyramidal cells that we have in the cerebral cortex. These pyramidal cells stain beautifully with this SMI32 stain. We see their cell bodies and their dendrites heading out towards the peel surface. And there is more space that is evident between the cell bodies that we see in the Nissl stain. That space isn't empty, it's actually filled up with synaptic connections. So layer 5 is a place where there is rich synaptic connectivity among its neurons. Well, just below layer 5, finally we have layer 6. Layer 6 in some respects resembles layer 4. There's a concentration of small cells that are more densely packed, sort of the way we have here in layer 4. And with respect to this SMI32 stain, layer 6 is, like layer 4, devoid of cellular staining. And finally, below layer 6, we have the white matter that underlies the cerebral cortex. Well, let's talk just a little bit more about how information flows through the cortical network. So I've already mentioned that our thalamus sends inputs up to layer 4, providing the main source of drive to this network of cells that populate the cerebral cortex. There's also a small input to layer 6, but the primary one that seems to have the greatest physiological impact is destined for layer 4. Well, from there, cells in layer 4 interconnect with one another, but they also send major inputs to the upper layers of the cerebral cortex, layers 2 and layer 3. And once information gets into those upper layers of cortex, it can be broadly distributed. So information can go in the horizontal direction from one region of layer 3, for example, to a neighboring region of layer 3. Information can be projected through the corpus callosum to the corresponding cortical area in the opposite hemisphere. So there can be long projections that come out of these neurons in layer 3 that enter the white matter and eventually the corpus callosum. One can also find local connections from these cells to nearby neurons within layer 2 and layer 3, as well as projections that will drive the activity of neurons down below in what we call our infragranular layers, those layers below the granular layer, layer 4, in layers 5 and 6. Well, those infragranular layers can give rise to descending projections. Layer 5 cells can project to a variety of structures in more inferior parts of the nervous system, including the basal ganglia, the brain stem, and even the spinal cord for certain regions of the cerebral cortex. Cells in layer 6 project back to the thalamus. They also provide a return input to layer 4 where this cortical circuit got started. Well, what I've been describing for you is the flow of information through the cortical microcircuit. And we believe that this basic flow of information, this basic circuit structure, exists across the entire cerebral cortex with some modifications depending upon the different cortical regions in question. But this basic flow of information of starting in layer 4, progressing to upper cortical layers, then down to lower cortical layers, is a common theme of cortical processing. Well, this circuitry performs a variety of functions. We're only beginning to scratch the surface to truly understand what those functions are. But I want to give you a sense of what the function is of this columnar circuit that we find in the radial dimension of the cerebral cortex. We call this the canonical microcircuit of the cerebral cortex. Well, I'm going to suggest an acronym that makes sense in this part of the world, and I hope it might make sense wherever you are. Well, if you know anything about Duke University, you know that our athletic department is pretty important to the life of this university, and one sport in particular seems to dominate, and that's basketball. Well, for those of you familiar with how basketball works in this part of the United States, you're familiar with 3 letters, ACC. But you know that as the name of the conference within which our Duke basketball teams compete. That is the Atlantic Coast Conference, the ACC. Well, I'm going to suggest that we hijack those letters and use it to remember some of the basic functions of this canonical microcircuit in the cerebral cortex. I would suggest that the ACC of the cortical microcircuit is amplification, computation, and communication. Let me tell you what I mean. Let's consider first amplification. So, as I've already described for you, the thalamus provides the principal input that modulates activity in cortical networks. That activity goes into the middle layers of the cerebral cortex, layer 4, and from there it's amplified. So one purpose of this network of radial connections is to amplify this signal. And once that signal is amplified, then new computations are possible. So the networks that we find largely emanating out of our upper layers, our supragranular layers, can take that amplified signal and now compute new properties that were not carried with that signal as it first entered the cortex. We'll see some examples of that when we get into our sensory systems. And then also from this part of the cortex, as well as from deeper layers, this information can be communicated broadly. And that communication can happen from one cortical column to the next, from one cortical area to another, and even from cortical areas to more distant regions that lie below the level of the cerebral cortex. So, amplification, computation, communication, the ACC of the cortex. Hopefully you'll be able to remember that. Okay, now I want to finally turn to the question of the organization across the extent of the cerebral cortex. And this allows us to return to what we looked at earlier, and that is the cellular architecture of the cerebral cortex. This is what we refer to as cortical cytoarchitecture. Cyto referring to cells, and architecture referring to the arrangement of those cells. So I would just remind you that neural tissue is comprised of a variety of different kinds of cells, big cells, small cells, from a physiological point of view, excitatory neurons, inhibitory neurons, those that fire in various kinds of patterns. Just a tremendously rich diversity of cell types are found in the cerebral cortex. And these cell types are organized in columns, but those columns have different compositions in different parts of the cerebral mantle. We see some of them illustrated here in this slide. So for example, if we look in the visual part of the brain, back here in the occipital lobe, we see an arrangement of cortex quite similar to what you saw earlier in this tutorial. In fact, those histological slides were taken from the visual cortex. And if we moved a little bit more anterior into the motor cortex, we'd see something that looks a little bit similar, but some things would really stand out. One thing that stands out is the presence of these really large cells in layer 5. These are called BET cells. They're the largest neurons we have in the cerebral cortex. But there are a little bit more subtle differences that you can appreciate once you have a bit of experience looking at the cerebral cortex. You'll notice that while the granular layer 4 is very prominent in our sensory cortex, it's largely diminished in its thickness as well as the density of cells in our motor cortex. That's because the function of the motor cortex is largely to provide output rather than to receive input. As we progress towards the more medial aspects of the brain, and the more ventral part of that cerebral mantle, we encounter a region of tissue that is wrapped into the medial edge of the temporal lobe. And this tissue is perhaps the most simple of all the regions that we find in the cerebral network. The anatomists have chosen not to recognize 6 layers as we do in what we call the neocortex, or that more phylogenetically modern part of the cerebral mantle. And what we find is an older part, a paleocortex, and even an archicortex. These are terms that we think reflects the evolutionary history of the cerebral mantle in the mammalian brain. Well, around the turn of the 19th to 20th century, European anatomists began to develop a variety of ways of studying the cellular architecture of the cerebral cortex. And one individual really stands out among many seminal contributors at the time. His name was Corbinian Brodmann. So Brodmann was a German scientist who studied the cytoarchitecture of the cerebral cortex, and he proposed a scheme for recognizing divisions of the cerebral cortex that is still with us today. We call these Brodmann's areas. That's what's illustrated here in this color-coded representation of the cerebral mantle with all of these numbers in place. So Brodmann was a fascinating figure, and it's a really interesting time in the development of modern neuroscience. So if anyone's interested, I would invite them to read a little bit more about Brodmann. And for the rest of you, you will invariably encounter his nomenclature as we talk about different parts of the cerebral mantle. So what Brodmann did was he looked under the microscope at a Nissl stain and recognized that different parts of the cerebral cortex have a different appearance, very much like what is shown here in this slide that I just showed you. And so Brodmann proposed, I think, a rather bold idea, and that is that these cytoarchitectural differences that he recognized in the cerebral cortex had functional consequences. And his scheme has largely ushered in the modern era of neuroscience, where we come to recognize that different parts of the cerebral mantle are specialized for certain kinds of function. Now, we can get a little bit carried away with this conceptualization of the cerebral cortex and have an erroneous view that cortical areas function in isolation. That would be a mistake, because different parts of the cortex maintain very specific interconnections with surrounding cortical areas, such that the cortex operates as an integrated network. But nevertheless, there are networks nested within networks, and the recognition of Brodmann's areas allows us to talk about those networks in ways that allow us to interpret our data that we're generating with, let's say, functional magnetic resonance imaging studies, as well as more invasive methods that can be done in animal model experiments where we're looking at cell-to-cell patterns of connectivity and function. Well, I hope you've enjoyed this brief survey of cortical structure and function. We will see application of these principles as we continue to explore our sensory and motor systems. And hopefully this concept of the ACC, of the cortical microcircuit, will be a useful way for you to understand what exactly does the cerebral cortex do in neural processing. Hi everyone, welcome to this tutorial on the general principles of sensory systems. We are at a bit of a pivot point in the course where our focus has been on gross brain anatomy as well as the principles of neurophysiology that underlie neural signaling and even a discussion of neuroplasticity. Now we're going to begin to bring these concepts together. On the one hand, the discussion of anatomy, and the other, the discussion of cellular physiology. And we want to bring them together as we discuss the organization and function of our sensory and motor systems. So this is the beginning of that pivot, and what I'd like for you to be able to focus on in this tutorial are some of the general principles that will help us understand the organization of our sensory systems, which is what we'll discuss first, and then we'll move on and discuss the motor systems. So this topic pertains once again to our key concept or our foundational core concept in the field of neuroscience pertaining to the complexity of the brain. And this tutorial, I trust, will help break down some of that complexity for you as we consider how sensory systems are organized and how they function. And this will allow us to maybe reflect for just a moment on this incredible capacity we have to explore our world, even the world within inside our own brain. You know, I'm reminded of a student I once had some years ago who I will not name in case you're out there somewhere, but he showed up one day to the lab and he said, you know, I just don't think the brain should be allowed to study itself. And I thought, what a profound observation. You know, the kidney doesn't get to study itself, nor does the liver, but somehow the brain has the credentials that allow it to study itself. And I think that is an interesting thought to consider. Well, let's move on and consider what we have to learn from this tutorial today. So I have several learning objectives for you. I want you to be able to account for the generation of action potentials in peripheral axons in response to somatic sensory information. Now, the somatic sensory system will be the first of the sensory systems that we consider, so I think it's useful to see how this general problem of action potential generation in a sensory system is realized in this one domain of sensation, that of somatic sensation. I want you to be able to account for differences in mechanosensory discrimination across the surface of the body. Again, we're going to take somatic sensation as our model paradigm, but the basic principles will apply to our other sensory systems as well. And I want to consider, in fairly general and broad terms, how information is coded in the nervous system. So I hope that you'd be able to have a discussion of the important factors that influence information coding. And one very important concept in sensory physiology is that of the receptive field. So I want you to have a thorough understanding of what the receptive field is, and one way to assure yourself of that is to have a discussion with someone about it. Okay, what I'd like to do next is to consider some general organizational principles that will apply to our consideration of our sensory systems. And we'll see how they're played out specifically in different systems over the next several tutorials, but today I'd like to keep it fairly general. So let's begin with one principle, and that is the principle of neuronal pathways. So let me illustrate for you in very simple terms what I mean by a neuronal pathway. Let's imagine that there's some kind of sensory surface out here. Perhaps this is the skin, and the skin is innervated by nerve fibers. And those nerve fibers are grown by cells, in this case cells that reside in the dorsal root ganglion. So an axon extends out and there's some kind of specialized receptor structure as the axon reaches the skin surface. So this is what we call the peripheral axon of this neuron. So this neuron can be considered a first order neuron in this pathway. Well, there is a central process that enters the nervous system. So let's say this is now into the central nervous system. And that first order neuron synapses on a second order neuron. And that second order neuron projects some distance through the central nervous system to typically a third neuron. We'll call that a third order neuron. And then that third order neuron provides input to a vast network of interconnected cells and some processing station within the brain. Perhaps this processing station is the cerebral cortex. So we might have some complicated network that allows all this to function. So we might call this now the cortex. So the neural pathway would be the first order neuron, the second order neuron, the third order neuron, and then once we get into our complex network that is being modulated by this ascending input, we sort of give up on our numbering scheme. So we'll see how that works. But the pathway, in essence, is a series of neurons that are connected. But the pathway is essentially a set of neurons that are connected in a serial fashion. And this allows neural signals to be generated at some receptor surface and then be transmitted into the nervous system, ultimately to be processed at the level of the cerebral cortex. Now, I drew this pathway in the horizontal dimension across the screen just to make use of the space that I have, but I want to make an important point about how we talk about our pathways in the brain. So let me clear that and redraw this in the vertical dimension. So often we have some receptor connected to our first order neuron, which projects to a second order neuron, but now we often think about this as ascending into the nervous system. And here's our complicated network again, representing the cerebral cortex. So here's our first order neuron, our second order neuron, our third order neuron, and then the cortex. Now this we might consider to be a sensory pathway, and the information is essentially flowing in the ascending direction. Well, this cortical network also has a motor component to it, where these sensory signals are eventually integrated within some kind of a network that coordinates and produces an output. And that output eventually reaches our motor neurons and behaviors result. So we can imagine that there's some output neuron here that is going to grow an axon, which will probably synapse on some kind of local circuit cell that modulates the output of a series of neurons that ultimately supplies some kind of effector, such as a skeletal muscle. So this now is an output pathway which can be conceptualized as descending from a higher level, from the cerebral cortex, for example, down to a lower level, maybe in the spinal cord, where we have a motor neuron leading to the contraction of a skeletal muscle. Okay, so pathways can be either ascending or descending. Ascending typically implies a sensory function. Descending implies a motor function. In our nervous systems that are bilaterally symmetrical, there's a very interesting principle of organization at play, and that is a principle of contralateral representation. So let me illustrate that for you. Imagine that we have some body part in question here. Maybe it's a upper extremity, and that upper extremity is going to be represented in the contralateral, or the opposite side of the cerebral hemisphere. So this is the cortex. Let's say this is the post-central gyrus, which is where we find our somatic sensory cortex. So this body part will find its representation in the somatic sensory cortex. Something like this. Now, contralateral representation implies that there must be some sort of crossing of the midline. Somehow this information has to get from one side of the body to the other in order for the ascending sensory pathway to relay it to the appropriate processing station at the level of the cortex. So as we work through our studies of our sensory pathways, please pay attention to this issue of decussation. So decussation simply means midline crossing. So we're talking about where in the nervous system does this pathway cross the midline so that the principle of contralateral representation can be understood. Now, this is not just an academic exercise. For future clinicians, this is a critical point. One of your challenges in the examination of your patients will be to understand, to have a mental model of what's wrong with the nervous system. There may be a focal lesion somewhere in this nervous system, and understanding where sensory and motor pathways cross the midline will be critical for your proper understanding of what level of the nervous system has been impaired. Now, this issue of decussation, it's not just for sensory systems, it's also for motor systems. Now, if we get just in front of this postcentral gyrus, we may be in the precentral gyrus, which is where we have our motor control networks, and there may be a neuron there in the precentral gyrus that gives rise to a descending axon. And that descending axon has to confront the same challenge of contralateral representation. At some point, there has to be a crossing of the midline, a decussation. And as it turns out, the decussations for sensory and motor pathways are not in exactly the same place. Even within our somatic sensory systems, different pathways cross the midline at different locations, and that will be critical for understanding the neurological signs and symptoms of injury and disease that you may observe in your patients. Now let's turn to a new topic, the topic of sensory transduction. By sensory transduction, what I mean is the process that allows for the energy that's present in the environment, either in physical or chemical form, to be converted into electrical signals within nerve cells. And sensory transduction is a problem for each of the sensory systems that must be solved in some ingenious way. And in fact, it often is. And many times there are highly conserved mechanisms that are employed across animals with nervous systems, including us. And sometimes those methods seem quite surprising. So I hope you have some fun discovering how sensory transduction works in each of the sensory systems that we will consider in this course. Let's consider one example from the somatic sensory system. Well, we know that the somatic sensory system is concerned with touch, with physical contact as one modality of stimulation, and it must happen that physical contact leads to the generation of electrical signals. So sensory transduction would be part of that story in addressing the question, well, how does this mechanical contact lead to the generation of electrical signals? Here's how this works for the receptors that are sensitive to mechanical deformation of the body surfaces. Imagine that we have some kind of receptor ending at the end of our first order neuron. This happens to be a beautifully laminated structure called a Pacinian corpuscle. And this Pacinian corpuscle contains a nerve ending that has cation selective channels. And these ion channels are sensitive to stretching forces that are applied to the membrane. So when there's some kind of mechanical interaction with the structures containing these Pacinian corpuscles, these ion channels will open. Not because they're voltage gated or because they are activated upon by neurotransmitters, as we saw in our previous tutorials on neurosignaling, but rather because the mechanical forces open these channels. So here's what we observe. When a physical force is applied to this tissue, these ion channels will open. This will allow our cations to enter. In this particular case, the principal permeant ion is a sodium ion, and so, as I think should now be familiar to you, as sodium rushes in to a neuronal cell, it carries with it its positive charge, and that leads to the depolarization of the sensory ending of this axon. Let's have a physiological perspective on this process now. Imagine that a fairly weak stimulus is applied, and that weak stimulus will lead to the influx of a relatively small number of sodium ions. So one can imagine a fairly weak depolarization of this receptor. Now, we have a term that we use to apply to this kind of depolarization. We call this a receptor potential because we're talking about a change in membrane potential in a receptor ending of a primary sensory axon. Now, if the strength of this stimulus is increased, what we would expect to see is a greater response. And indeed, that's what we see. So with the stronger stimulus, we see an increase in the amplitude of this receptor potential. But notice that we don't yet generate an electrical signal that can propagate as long as this potential stays below threshold for generating an action potential. But with a stronger stimulus, it very well may be that this receptor potential is now of sufficient magnitude to achieve the threshold for generating an action potential. So once that threshold is achieved, an action potential fires. Now, notice what's going on here. The generator potential is a function of the stretch-activated ion channels. But once that membrane is depolarized to achieve the threshold for generating an action potential, now our friends, the voltage-gated sodium and potassium channels, are activated and an action potential is generated. So different kinds of ion channels contribute in different phases to the transmission of information along the axon of the primary sensory neuron. Now, in the case of our subthreshold potential, our receptor potential that failed to elicit an action potential, we're never going to know about that contact through this sensory ending because there has been no action potential. There's been no signal transmitted into the central nervous system. That requires a receptor potential of significant amplitude to achieve threshold. Now I want to turn to the topic of information coding. And one concept that we'll see played out in the somatic sensory system, in the visual system, in the auditory system, in our chemical sensory systems, and even in our vestibular system, is the notion of labeled lines. Now, what we mean by labeled lines is the idea that the response of an axon in a sensory nerve is going to be shaped or dictated, if you will, by the kind of receptor that's elaborated at the end of that axon. And that sensory receptor is going to be specialized to encode a certain quality of information. So what we're looking at here is a cross-section through the skin, and what we see is a rich variety of sensory specializations. So what we're looking at are a variety of endings of the axons of our first-order neurons. Many of these are going to be sensitive to different aspects of mechanical stimulation, but some of them are going to be sensitive to the potential threat to damage of the skin in relationship to injury, to temperature change, to the presence of chemicals that might harm the skin. So these are examples of what we have in mind by labeled line. And here's one example in particular. Here's that Pacinian corpuscle that we illustrated schematically a moment ago. This structure is found deep in the skin, in the subcutaneous layers or near the deeper part of the dermis, and this is a structure that's especially sensitive to vibration. Meanwhile, there is a type of receptor that is right where the epidermis and the dermis interdigitate. It's called the Meissner's corpuscle. This structure is especially sensitive to light touch, the kind of touch that we might use as we use our skin surfaces on our fingertips to explore a sensory surface. So the selectivity of the different axons that terminate in these receptors is really determined by the specializations we see at their terminal endings. And the fact that different axons are associated with different types of receptors implies that there are parallel channels that transmit various submodalities of sensation from the peripheral surface back towards the central nervous system. So another principle that we see illustrated here in this figure is that of parallel processing. All of these afferent axons are transmitting information in parallel from their various locations in the skin into the central nervous system. In this case, either the spinal cord or the brainstem. Now let's consider how information is encoded in the firing patterns of neurons. And then this brings us to an important phenomenon that's present across all of our sensory systems, and that is adaptation. What we find is that when a stimulus is applied to a sensory ending, there is typically a barrage of action potentials that we see at the onset of that stimulus. But notice that over time, the numbers of action potentials typically diminishes. For some neurons, the barrage of action potentials is really all that we see. and after the initial onset of the stimulus, there's basically no further activity in that first order axon. Others continue to fire, perhaps with some regularity, but often at a diminished rate to the abrupt onset of that stimulus. So these are examples of what we call adaptation. And adaptation can happen very quickly and dramatically, or it can occur more slowly and to a less significant extent. So some receptors adapt rapidly and others adapt rather slowly, and this has consequence for the communication of information from our sensory surfaces into our nervous system. You can imagine that the slowly adapting types of receptors might be very useful for telling us about the persistent presence of a stimulus, whereas the rapidly adapting sorts of receptors might be quite good at telling us about the dynamic changes in the presence of a stimulus. Now, we've already considered the concept of threshold, and let me just make one further comment about it now. And that is that while all sensory receptors have some kind of threshold for generating an action potential, that threshold might be different for different kinds of receptors based on the sensory transduction mechanism that's involved, the specializations that may surround that nerve ending, and also with respect to the location of that receptor within the sensory structure, be it the skin or a muscle or a joint or wherever it may be. So sensory thresholds can be an interesting way that the nervous system can use to be sensitive to the right kind of information in just the right sort of way. So as an example, our mechanosensors, those that are sensitive to mechanical stimulation, tend to be very sensitive with a low threshold for activation, whereas our pain fibers, those associated with the free nerve endings in the tissues of our body, tend to have a higher threshold for activation. And that's a good thing, is it not? Because it requires further stimulation of our sensory surfaces in order to elicit pain. We wouldn't want a light, gentle caress to be painful. And by modifying the sensory thresholds of different kinds of labeled lines, we can achieve that goal. Lastly, I want to consider with you the topic of receptive fields. Now, this is a fascinating topic that we'll see over and over again as we consider our different sensory systems. And let's again stick for now with the example of our somatic sensory system. What we mean by a receptive field is simply that region of the body or that specific area in the space around the body that when a stimulus is presented, activity can be modulated in a specific nerve cell. And here's an example of how this might work. Imagine that there is a microelectrode inserted into the postcentral gyrus. In this case, let's imagine that we have a primate as an animal model, and we can record the activity of neurons in that postcentral gyrus. Now, if that should be the case, what we will discover is that that neuron requires stimulation of a very specific location in the body in order to have its activity modulated. And the job of the experimenter in this scenario is to discover what is the surface of the body that must be stimulated in order to modulate the activity of that neuron in the postcentral gyrus. And what the experimenter might discover is that there's some patch of skin that when stimulated can modulate the firing pattern of that neuron. So what we see here is that during the period of stimulation, there's an increase in the number of action potentials that neuron fires compared to what we see before and after. And furthermore, what we find is that receptive fields tend to have a center-surround organization. And here's what we mean by that. There tends to be a center of the receptive field that, when stimulated, gives rise to a robust modulation of activity. Often it's a dramatic increase in activity, which is what we see here, but sometimes it's actually a decrease in activity. That really doesn't matter. The important point is that there is a rapid modulation of nerve cell firing when the center of the receptive field is stimulated. Well, receptive fields are surrounded typically by an annular region that we call the surround. And the physiology of the center-surround interaction is antagonistic. That is, if the center of the receptive field leads to an increase in firing, then activation of the surrounding annulus typically leads to a decrement in firing, which is what we find for the patch of skin surrounding this particular receptive field on the skin for this cortical neuron in question. So notice how activation of the center leads to an increase in firing, whereas activation of the surround leads to a decrease in firing. We'll see the very same pattern of activation in the retina, in the visual parts of the thalamus, and in the early parts of the primary visual cortex when we consider the physiology of the visual system. Allow me to make a few additional points about receptive fields before we conclude this tutorial. Well, we've already looked into the dermis and appreciated the variety of receptor endings that we find there in different parts of the dermis. I'd like to point out that some of these receptors are really quite spatially limited in their extent, whereas others, because of their size and their organization, tend to extend over a greater extent. Well, as it turns out, there is, in fact, a variety of sizes of physiological receptive fields. So, for example, we can imagine that for this Meissner's corpuscle, perhaps we have to be right on top of it in order to stimulate it, whereas this Purkinjean corpuscle perhaps can be activated through deformation of skin over a much broader area. So the size, the orientation, the distribution of the peripheral sensory structure will have a lot to do with defining the size of the receptive field for the first order neuron. But we can actually record a receptive field for any neuron. As we saw in the previous slide, that receptive field was defined for a cortical neuron. So what we'll find is that at each level along a sensory pathway, there will be a variety of receptive field sizes and shapes that can be identified. For the first order neuron, the size is often related to these geometrical factors that I've just outlined for you. But when it comes to neurons within the central nervous system, the second order neurons, the third order neurons, the neurons in cortical networks, principles of convergence have much to do with defining the size of those receptive fields. So for neurons in the cerebral cortex, for example, what really determines the size of the receptive fields will be the degree of convergence. And for neurons in the cortex, large receptive fields are created by a high degree of convergence, that is, many neurons at lower levels of the sensory pathway converging onto the same cortical neuron, whereas smaller receptive fields are created by minimal patterns of convergence, that is, fewer numbers of cells at antecedent levels of a neural pathway converging as information is transmitted onto the level of the cerebral cortex. So at the level of the cortex, we see more of a direct relationship between receptive field size and the degree of convergence. The more there is convergence, the larger we would expect to see the receptive field. Now let's look at one final example from the somatic sensory system and how this plays out. Imagine we're going to do an experiment to assess two-point discrimination. And I would encourage you to do this with a friend or a family member. All you need is, let's say, a paper clip or maybe a little piece of wire that can somehow be bent into a shape, something like this. And you can vary the distance between the ends of this wire in such a way that maybe the wire can be bent so that these points of contact can be spatially varied. Well, what you can do is you can ask a friend or have them ask you to close your eyes while some modest part of your skin surface is explored with this kind of a homemade caliper. Well, if you do that, you can ask your friend, or your friend can ask you with eyes closed, simply tell me whether you feel 1 point of contact or 2. So 1 or 2, that's the question. And what you can find is what is the minimal separation of the tips of these calipers that will allow your test subject to report 2 points of contact. So this is called two-point discrimination. And you can imagine how this might work. In order for a subject to report 2 points of contact, we must stimulate at least 2 receptive fields on the skin surface. So this is illustrated for the fingertip and in a patch of skin in the forearm. And what is shown in this figure is what's actually observed in most people. That is, the receptive fields are smaller in the fingertips, where spatial acuity is quite high, compared to the forearm, where spatial acuity is much lower. And this means that 2-point discrimination will be better in the digit surfaces compared to the skin on the forearm. That is, we can detect smaller separations on our fingertips compared to our forearm. So from a neurophysiological perspective, let's consider these 3 examples. So here in green, we have 2 points of contact that are coming together quite closely within the receptive fields of neuron B. And what we see there is that with the green contact, neuron B is activated to a significant extent, but these points of contact only touch the margins of neurons A and C, so these neurons fire at not a very high rate. So for the set of primary afferents coming from this patch of skin, it looks like only 1 neuron was activated, and we might imagine that we would interpret that as 1 point of contact, when in fact there were 2 points of contact. Now let's look at the points of contact shown here in red, a little bit further separation. So we are now getting closer to the center of activating neurons A and C, but we're not yet so far to the extreme of the receptive field of neuron B that it's not going to be activated. So what we have as a result is activation of 3 receptive fields to a pretty similar extent. Well, this might also feel like just 1 point of contact, because we're not adequately differentiating the responses of one receptive field or one neuron from another. That only happens once we expand our caliper to the points of contact illustrated here in blue. We are very near the center of the receptive fields of neurons A and neuron C, and we're just at the margin of the receptive field of neuron B. So we would expect a robust response from neurons A and C, minimal response in neuron B. So in the context of a neural structure that's representing the information coming from these 3 parallel channels, we see clear differentiation of activity between 2 of these 3 neurons, with a separation between those most active neurons. This will allow us to make a judgment of 2 points of contact rather than 1. So we think this is the basis of 2-point discrimination. And indeed, if we survey the body surfaces, we find that there is a tremendous range of sensitivity. So what we're looking at is a plot of the 2-point discrimination threshold across the left and right sides of a subject. And what we find is really high sensitivity around the fingertips and some of the oral facial structures, and not so much sensitivity in some of the more proximal parts of our arms and our legs. Well, these concepts are illustrated here with respect to the somatic sensory system, but we'll see how they play out in other domains. I hope you appreciate that what we're talking about here with 2-point discrimination is really the phenomenon of acuity. So what we're talking about here is spatial acuity, but the same principles will apply, let's say, in the visual system, where we have very small receptive fields and a high density of receptors in the central part of the retinas in our eyes. That's a region of greatest spatial acuity. Whereas in the peripheral parts of our retina, we have larger receptors that are much less dense in their distribution. Consequently, we have much larger receptive fields in the neurons that are receiving input from that receptor surface. So acuity is an important concept in sensory systems, and we'll return to that as we go along in our studies. Well, now that I've given you an overview of some of the important principles of organization and function in sensory systems, I'll just leave you with the study question and give you an opportunity to consider that on your own. Welcome back to Medical Neuroscience. In this tutorial, I'd like to talk to you about the neural mechanisms for mechanosensation. This topic relates to really half of the core concepts in the field of neuroscience, which gives you a sense of really what the breadth of this topic is and how it might apply. So I would suggest that once again, as we understand how our sensory systems work, and certainly mechanosensation is a great example of how our sensory systems work, we are again confronted with the complexity of the brain in ways that make it really different from studying any other organ within the human body. We are going to be learning about circuits in the brain, circuits that connect different parts of the brain, and many of them are based upon the genetic instructions that are unfolding in the course of early brain development. So there are genetically determined circuits that are the foundations for the nervous system, and our sensory systems certainly are an important part of that foundation. Now, as we'll see in later sessions, that's not to say that they're not subject to use-dependent plasticity. They certainly are, but the foundation of those circuits is largely based on genetic information. Well, because of such circuits in the brain, our brain provides us with this just fantastic capacity for curiosity, and I hope that we exercise that capacity well as we approach our studies of our sensory and motor systems over the next several tutorial sessions. And then lastly, we'll increasingly apply our knowledge of sensory and motor systems to gain some insight into the diseased or the damaged nervous system. So I hope you'll see how fundamental knowledge, fundamental discovery about the brain can be used to help people who have been injured or have suffered affliction in their central nervous system. Well, we have several learning objectives today. So I want you to be able to identify and to characterize the major sensory endings that mediate sensations that are elicited by touch, by vibration, by proprioception. These are all elements of mechanosensation. And I will introduce the receptors for pain and temperature, although we will focus on those in a subsequent tutorial. I want you to be able to understand the concept of somatotopy, that is, of how the body is mapped in the structure of the nervous system. And so we'll confront that in the primary somatic sensory cortex in this tutorial. And I want you to be able to discuss what happens to mechanosensory information as it enters cortical networks. We will spend a fair amount of our attention focused on the organization of the postcentral gyrus, but I want you to have a sense of where does that information go from there. So that will come up at the end of the tutorial today. Okay, well, let's get started by considering just some broad organizational concepts as they will play out in the mechanosensory systems. Beginning with this notion of there being multiple parallel pathways that mediate different aspects of somatic sensation. So we're going to focus today on our mechanosensory pathways. And even within our mechanosensory pathways, we'll see this principle of parallel organization played out. We'll leave for another session, as I mentioned just a moment ago, consideration of our pain and temperature systems. Okay, well, speaking broadly about these two subsystems within our somatic sensory nervous system, let's consider some of their basic functions. On the mechanosensory side, one important basic function is that the mechanosensory system helps us to identify the shape of objects that we encounter, as well as the texture of surfaces that we might experience with our skin, especially the very sensitive surfaces on our hands and in our face. So one very important means we have to explore the environment will be with our somatic sensory sensitive surfaces. A very important role played by our pain and temperature systems is that these systems help to inform us about potentially threatening circumstances that our bodies might encounter in the environment. So, well, some of us might wish that pain would go away at times. Pain actually is very adaptive. It informs us about the potential threat that might be done to the body, or the occurrence of injury and the potential for further exacerbation of that injury. So these pain pathways are very important for moderating our behavior with respect to the world around us. Another important aspect of mechanosensation is that it helps us to monitor the forces that are acting upon the body. Some of those are internal. They reflect the activities of the body itself, largely the activities of our musculoskeletal system. But we also monitor the forces acting on the outside of the body, especially in certain activities such as sport, where we are encountering physical objects, and it's, of course, critically important for success in those activities that we be able to monitor those interactions and respond accordingly. And then lastly, I would suggest that all of these somatic sensory systems, our mechanosensory systems, and even our pain and temperature system, help to build up in our brain a image of our body, a sense of self, a self-awareness. We call this proprioception. And this is tremendously important, and it's something that we often take for granted. Somewhere within our brain, we have an image of our body. And in one hemisphere, we have an image of the opposite side of the body. And somehow between the two hemispheres, we integrate this into a united body schema. Well, as you might expect, that body schema is built on a steady stream of information flowing through our somatic sensory systems. And that stream is then met with information coming from other modalities of sensation, such as vision, audition. I would suggest even our chemical senses help to inform us about our body in the world around us. So it's the confluence of this information integrated at the level of the cerebral cortex with loops through deeper structures in the brain that give rise to this integrated sense of self. So we will begin a conversation about what that means today when we consider our mechanosensory pathways. Now, let me just consider a couple of points about the basic organization of our somatic sensory systems, beginning with this notion of labeled lines. I've talked about this in a different tutorial, so I won't repeat all of what I said there, but let me just remind you that the pathways for somatic sensation, as with our other sensory systems, are arranged by having a series of parallel channels that reflect specializations that begin right at the level of the receptor endings of the primary axons out there in the body, wherever they may be, in the skin surface or perhaps in deeper structures such as muscles and joints. We call this a labeled line system because the channel is labeled with respect to the kind of information that it carries. And that label really begins with the structure and the physiology of the receptor that's at the end of the primary axon. So we'll see that as we go through this. So keep that thought in mind about labeled lines as one organizational principle in sensory systems. Another basic feature of the organization of the somatic sensory systems is that we have multiple pathways that are spatially segregated within the central nervous system. So if there are labeled lines, you might wonder, well, where are those lines as they run through the CNS? And that's the question we're going to address as we go through this discussion of our somatic sensory systems, including the anatomy of those systems. And what we will discover is that there are two pairs of pathways that serve somatic sensation. Really, they can be further subdivided, but we'll keep it pretty broad and simple and say there are two pairs of pathways. There's a pair of pathways for the body, including the back of the head and on down below the head. And then there are a pair of pathways for the face. As you might imagine, the face is a very important part of our body with respect to somatic sensation. And for reasons that aren't entirely clear, there is a dedication of considerable brain space to the processing of signals that arise from the face in a manner that's similar to the dedication of brain space that is involved with processing signals that are derived from our hands. So we'll provide a partial explanation for why that might be so as we go along today. Okay, so we won't go into the detailed anatomy of these pathways in this tutorial. We will reserve that to dedicated tutorials that will follow. Okay, now as we begin to consider the organization of these systems, let's focus in on the first order neuron. And our attention will be turned primarily to the mechanosensory systems, but as we'll see, for completeness, I also want you to see some of the differences between these labeled lines for mechanosensation and for pain and temperature. All right, so let's consider the general organization of this first order neuron. So the first order neuron in our pathways for somatic sensation are dorsal root ganglion cells. And so what's illustrated here is the connection between the sensory signals that arise in the fingertip and the central nervous system. So these dorsal root ganglion cells, they have cell bodies that reside in this dorsal root ganglion. So this is a home for cell bodies associated with the dorsal root that attaches to the spinal cord. And these cells have a very distinctive histological morphology. These cells are pseudo-monopolar, and that means that there's a cell body, it grows out an axon that then bifurcates and grows in two directions. One direction grows out to the body, and the other grows out to the spinal cord. So that's what we mean by a pseudo-monopolar neuron. That's what we find in the dorsal root ganglia. So the peripheral process of this cell grows out to reach the surface for which it is going to serve. And in the case of a mechanosensory fiber, which is what's illustrated here in red, there may be some kind of specialized receptor ending that we'll see in just a moment that will begin to label that line as being a mechanosensory axon. Well, we also show a different cell here, one illustrated in blue, that is going to be sensitive to pain and temperature signals. And we'll talk more about this pathway in a later session. What we'll find is that there is a different kind of receptor ending there and a different set of proteins expressed on that ending that label that line as being sensitive to pain and temperature. Well, that's the peripheral process. What about the central process? One hint at where we're going to go with our studies of the anatomy here is that the central process of these two different kinds of dorsal root ganglia do very different things as they enter the dorsal root entry zone of the spinal cord. The mechanosensory axon enters that dorsal root, and notice that it makes a sharp upward bend. In truth, there are other branches that do other things, but we're going to ignore those for the time being. Rather, I want to emphasize to you that there is a growth pattern for the mechanosensory fiber that's really distinct. This axon enters the dorsal column of the spinal cord, the white matter that we call the dorsal column. Now contrast that with the pain and temperature fiber. Its central process enters the dorsal horn and makes a synaptic connection right away. So here we have a synapse with a dorsal horn neuron. And that dorsal horn neuron then grows out an axon that does something pretty interesting that we'll talk about later. Okay, so one clear difference between these two labeled lines is that the mechanosensory fiber grows along the course of the spinal cord, whereas the pain and temperature fiber makes a synaptic connection upon entrance into the spinal cord at the level of entrance. Okay, so keep that thought in mind, and we'll see how that plays out when we talk in detail about the pathways. Okay, a little bit more about these first-order neurons. These first-order neurons can be organized in various ways, and this table from our reading from Chapter 9 gives you probably more information than you need to know, but at least it makes clear a few basic principles, and that is that there are different morphological categories of first-order axons that relate to their functional properties. For example, you'll notice that the receptors that are going to be sensitive to the movements of our body, we call those proprioceptors. One example would be a muscle spindle. These receptors are supplied by really large, well-myelinated axons. There's a name for them. They're called group 1A, group 2 axons. Now contrast that to the receptor endings that are going to be sensitive to pain and temperature. Those would be down here. So these have a different morphological ending. We'll see that in just a moment. They're called free nerve endings. And these free nerve endings are formed at the ends of our smallest axons, our smallest sensory axons that we have in peripheral nerves, some of which are very lightly myelinated. We call those A delta, and others are barely myelinated or completely unmyelinated, and those are called class C fibers. So group 1, group 2 fibers, these are very large fibers. Group C is our smallest, and A delta is very close to that. There is a group of mechanosensory fibers that are sort of in the middle, but they're much more like the group 1 fibers. These are sensitive to various aspects of mechanosensation in our skin surfaces, and we'll talk about these receptors in just a moment. These axons are also pretty hefty with lots of myelin around them. They're just not the fastest of our axons with respect to conduction velocity that we have. So we see how these fall out here on this chart. Okay, so now let's consider the concept of sensory transduction. So you've already been introduced to this idea. This is the concept of how is the energy of the environment converted into electrical signals. Well, for the mechanosensory system, this happens when physical deformation of ion channels leads to the opening of those channels and the influx of current-carrying ions. So for our somatic sensory system, this happens as channels that are permeable to sodium ions are forced open. So the physical pressure forces open these ion channels. They are activated by physical forces, not by voltage, not by neurotransmitters, but by physical forces here. And that leads to the influx of sodium ions. And so as these sodium ions enter, as you should now appreciate, they carry with them their positive charge, and that leads to some measure of depolarization. So if you open a small number of these sodium channels, you're going to end up with a small depolarization of that axon terminal. So this depolarization is called a receptor potential because it originates with some kind of receptor element at the end of the sensory axon. If you provide a stronger stimulus, you'll get an even stronger receptor potential. But if that potential fails to reach threshold for generating an action potential, this signal will simply decay away, and you'll never know it because there will not be an electrical signal that can propagate from the receptor ending into the central nervous system. That only happens when the stimulus is sufficiently strong to reach the threshold for generating an action potential. So once that receptor potential hits threshold, now our familiar voltage-gated ion channels kick in, and we end up firing an action potential. So the stretch-gated channels, they get us to threshold. That's the receptor potential. Sometimes we call that a generator potential. But the firing of an action potential is where our voltage-gated sodium and potassium channels come in. That's what gets us from a receptor potential in the end of a sensory axon all the way into the nervous system with the potential of actually experiencing that sensation if the signal makes it into the cerebral cortex. I've been alluding to the morphological specializations at the end of our mechanosensory fibers. Now let's have a look at them. And we see really a beautiful array of specializations, even within our skin surfaces. So what we see depicted in this figure is an illustration of our endings of these sensory axons and what happens to them. Let me first highlight those receptors that are responsible for our sense of light touch. So go ahead and rub your fingers together just very lightly, or perhaps pass your fingertips over a tabletop surface that you may be near. And that very light touch sensation that you may be feeling on your skin surfaces, that's due to the activation of Meissner's corpuscles and our Merkel cell neurite complex. These are both exquisitely sensitive to mechanical deformation, but they operate in slightly different ways. The Meissner's corpuscle is a specialization of the nerve ending with Schwann cells that form a layered structure and constitute what we call a corpuscle. This is found as the dermis interdigitates with the epidermis, and we find these structures oriented in sort of a radial direction relative to the skin surface. That makes them acutely sensitive to a small patch of skin that might be deformed. Now contrast that with our Merkel cell neurite complex. This is a specialized arrangement of nerve endings with cells that sit between these dermal papillae. And there's actually neurotransmission that occurs here. So a receptor potential is going to be generated in these specialized Merkel cells. A neurotransmitter will be released, and that will activate the nerve terminal of the first order axon. So together, the Meissner corpuscle and the Merkel cell neurite complex are responsible for most of what we think about as light touch signals. Okay? So you'll notice that there are some odd looking receptors that are found a little bit deeper in the skin, in the dermis and on into the subcutaneous layer. These are two different kinds of encapsulated receptors. One is called the Ruffini corpuscle, and this is a rather strange structure. We actually don't understand its physiology quite as well as some of the others, but it seems to be sensitive to stretch. So if you were to extend the digits in your fingers and sort of wave out, fan out your fingers, you're stretching those skin surfaces, and that sense of tugging in your skin is probably derived from these Ruffini corpuscles. And then finally, deeper in the dermis or in the subcutaneous tissues, we have these beautiful structures called Pacinian corpuscles. So this is a nerve ending that's encapsulated with many layers of Schwann cell lamellae, and it's almost like the layers of an onion around a nerve ending. And these layers are buffered with a fluid, an extracellular fluid, and so what happens is that as there's some depression, there's a very quick and rapid activation of that sensory ending, but very quickly the fluids redistribute and that deformation rebounds, and we end up restoring very quickly this rings of onion type of structure around that nerve ending. So these are endings that are going to be sensitive to vibration, but they're going to act only very phasically as soon as that deformation is applied. Well, to speak to the physiology of these various receptors, I'm going to show you a table from the book which truly has more information than I want you to know. So let me just tell you what about this table I think is important for you to take away. I certainly want you to recognize our four major types of superficial receptors that we've been talking about. And I want you to notice that there are two, our Merkel and Meissner structures that have small receptive fields, which means they're going to be especially sensitive to spatial position. And then there are two, those that are deeper, our Pacinian and Ruffini corpuscles that have larger receptive fields. So pay attention to the extent of skin surface that can activate that receptor. I would also invite you to focus in on the function that's listed here in this table. So notice that the Merkel and Meissners have to do with light touch and perhaps even different aspects of light touch, whereas our deeper receptors have to do with broader sensations that have less spatial detail in them. In the case of the Pacinian, mainly sensitivity to vibration, and as I mentioned for Ruffini corpuscles, a change in shape that would be consistent with a stretch of the skin. And lastly, I would encourage you to pay attention to the innervation density, which relates to the spatial acuity that is provided by this receptor system. Notice that the innervation density is quite high for our Merkel cell neurite complex, and really extremely high for our Meissner's corpuscle. That contributes to the pretty high spatial acuity of this system. Whereas our deeper receptors are Purkinje corpuscles, and our Ruffini corpuscles are present at much lower density. And that's consistent with them being sensitive to a broader surface of the skin. Okay, let's move on and let's consider some additional types of receptors that are found in deeper tissues in the body. One very important proprioceptor that we'll talk about in some detail when we talk about spinal cord circuits for motor control is something called the muscle spindle. And so the muscle spindle is formed by specialized muscle fibers and certain types of receptor endings that innervate those specialized muscle fibers. So what we see here is an illustration of a muscle. So this is a muscle that is part of a musculoskeletal unit, and within that muscle we find a fusiform shaped spindle that is bounded by a capsule. So within that spindle we find a small number of muscle fibers. Those muscle fibers are called intrafusal muscle fibers, and they're different than the muscle fibers that are found outside this spindle that generate the force when we contract a skeletal muscle. These muscle fibers tend to have a collection of nuclei that are bundled near the center of this spindle shape, and then they have their contractile elements extending away from that central region out to the pole of this fusiform shape. And these two regions of the muscle spindle are supplied by two different kinds of sensory structures. There is a type of sensory axon called the group 1A afferent that innervates this central region of the muscle spindle, this bulbous part. And notice how it does so. These axons just wrap around the central region of these muscle spindles. There's a second type of sensory axon that supplies the muscle spindle. It's called a group 2 afferent axon. And rather than wrapping around the central portion of the intrafusal fiber, this axon terminates in a special type of ending that we call a flower spray ending. And it's located on the contractile elements of the intrafusal muscle fiber, extending some distance away from this central bulbous region of the spindle. Taken together, these two kinds of spindle afferents, the group 1A and the group 2, allow this muscle spindle to send signals into the spinal cord that are pertaining to stretch. So as the muscle is stretched either actively or passively, basically this spindle gets stretched out as well. And that spindle ends up putting mechanical pressure on these sensory endings that are found at the terminals of the group 1A afferents as well as in these flower spray endings associated with the group 2. So this makes these sensory signals important for determining the length of a muscle because they're sensitive to the stretch that's being applied. We'll come back and talk more about that later. But let's move now to the last type of sensory receptor that I want to show you. This is also associated with skeletal muscle, but rather than a axon supplying the muscle fiber directly, this is a axon. We call this a group 1B afferent, and it innervates the junction of the muscle fiber and the tendon. So there's a capsular region between the tendon and the muscle fiber, which is made of a woven matrix of collagen fibers. And basically what happens, as is detailed down below, is the axon branches and interdigitates within these collagen fibers. And as a result, when there is a force applied to this tendon as these extrafusal muscle fibers contract, this collagen matrix gets stretched. And that matrix, because of the interdigitating axons, puts pressure on those axon terminals. And so that is the force that opens the ion channels, leading to the generation of receptor potentials in these nerve endings. So this type of receptor, it's called the Golgi tendon organ. This type of receptor is going to be sensitive to muscle force rather than to a stretch being applied to the muscle. We'll talk more about these receptors when we get into the circuitry of the spinal cord and how muscle tone is regulated. But for now, just know that there are two kinds of receptor systems associated with our musculoskeletal units. There are Golgi tendon organs that are sensitive to the force that's generated by the contraction of a skeletal muscle. And there are muscle spindles that are going to report the length of that muscle back to the central nervous system. Not the force, but the length. And so the relevant stimulus for the muscle spindle is stretch. Now lastly, I'll just mention, but I won't show you, that there are also receptors associated with our joints that inform the nervous system about the movements of those joints or the pressures that are acting upon them. So taken together with the muscle spindles and the Golgi tendon organs, we can think of these as being the proper proprioceptors that inform the central nervous system about the movements of the musculoskeletal system. Okay, so we've been talking about our first order neurons and their organization in the periphery and just a little bit about their organization as they enter the central nervous system. We're going to devote an entire tutorial to following the pathway by which the first order neuron eventually communicates with higher structures in the forebrain. So we won't look at those details now, but I do want to jump to identifying what are those centers that are involved in processing mechanosensation in the forebrain and give you just a little bit about their physiology. So let's begin with talking about the somatic sensory division of the thalamus. So there's a brief tutorial that talks about the thalamus to get you oriented to this part of the forebrain. For now, I'll just remind you that the thalamus is derived from the diencephalon, which is part of the forebrain of the prosencephalon, and it's the more dorsal posterior part of the diencephalon, with the hypothalamus being more ventral and anterior. Well, the thalamus, as my other tutorial goes into in more detail, is divided into a number of nuclei. And those nuclei relate to a particular part of the cerebral cortex. So if we were to look at the cerebral cortex, we could identify just about any region that we may decide to poke at and ask, what is the thalamic nucleus that projects to this region of the cerebral cortex? And we'll have a different answer for most different sites in the cerebral mantle that we might inquire about. Well, if we focus on the postcentral gyrus, which is right here, that's, as we'll talk about in just a moment, is our primary somatic sensory cortex. There's a particular part of the thalamus that maintains interconnections with this postcentral gyrus. That part is exactly the part of the thalamus that receives the ascending information about mechanosensation. That part is what we might call the somatic sensory thalamus, and we have some anatomical names to give it. We call it the ventral posterior complex of the thalamus. And that ventral posterior complex is divided into two main nuclei. There's a division called the ventral posterior lateral nucleus and a division called the ventral posterior medial nucleus. And the division there basically reflects the terminations of our pairs of pathways that are representing the body from the posterior part of the head on down, and then a different pathway conveying mechanosensation for the face. So for the body below the back of the head, we have signals arriving in the ventral posterior lateral nucleus, whereas signals concerning mechanosensory activation of facial structures arrive in our ventral posterior medial nucleus. So now we're ready to consider what happens to this somatic sensory information as the thalamus communicates it up to the level of the cerebral cortex. And that gets us into our postcentral gyrus. So here once again is the postcentral gyrus in my model of the human brain here. And this is where we're going to find the region of cortex that we call the primary somatic sensory cortex. The reason why it's primary is that it's the first part of the cortex to receive input about mechanosensory information. And in anatomical terms, it's the part of the cortex that receives input from the somatic sensory thalamus. That is the ventral posterior lateral and ventral posterior medial nuclei. And as we'll see, the ventral posterior lateral nucleus projects from the paracentral lobule in the midline of the hemisphere along this postcentral gyrus as it extends across the dorsal and lateral aspects of the cerebral hemisphere until we get to about 2/3 the distance between the midline and the lateral fissure. When we get to about this region, now the rest of the postcentral gyrus is receiving input not from the VPL, but from the VPM, the ventral posterior medial nucleus. This explains why in the mapping of the body that we will see in just a moment, there's a disjunction between the representation of the hand and the representation of the face. It's basically the boundary between the projection zone from the VPL and the VPM. Okay, well, let's look at this now in a bit more detail. So here we have an illustration from your text which shows us our primary somatic sensory cortex, which we call S1 for short. So it's found in the postcentral gyrus, and it's comprised of 4 cytoarchitectonically distinct regions of the human brain that Corbinian and Brodmann named over 100 years ago areas 2, 1, and area 3. And workers after Brodmann subdivided area 3 into an area 3A and an area 3B. So from posterior to anterior, there are areas 2, 1, 3B, and then 3A right at the depth of the central sulcus. So in each of these cortical areas, we have a complete representation of the opposite side of the body. And that representation is beautifully mapped out. We call this somatotopy, which means body mapping. And the way the body mapping works is that the distal part of the lower extremity is represented in the midline, right at the medial termination of the postcentral gyrus in this region that we call the paracentral lobule. So this is essentially where we find the representation of the contralateral foot. And as we progress over the dorsal rim of the hemisphere and onto the dorsolateral surface of the postcentral gyrus, the body map progresses from the foot up the lower extremity and then into the region of the trunk. And then from the region of the trunk, we encounter the shoulder, and then we get into the upper extremity. And where the central sulcus has a fairly distinctive curve in it, that's the region where we find an expansive representation of the contralateral hand. Now, notice we have not yet encountered the face. That's because, as I just mentioned, the face is conveyed by inputs from the ventral posterior medial nucleus of the thalamus, which terminates in this inferior segment of the postcentral gyrus. So just below this S-shaped bend in the central sulcus, we have a junction between the representation of the hand and the representation of the face. The illustration that we have in front of us shows us the detail of that body mapping, and it truly is an exquisite mapping of the body, with the progression from lower extremity through the upper extremity, and then a disjunction where we then have a break and the representation of the face in the inferior third or so of the postcentral gyrus. Now, there's a wonderful story about a famous neurosurgeon at the Montreal Neurological Institute, Wilder Penfield, and his assistant. and there's some discussion as to how much of this is really true or not, but evidently the assistant is the one that suggested to Dr. Penfield that this distorted representation of the human body be represented in a simple cartoon. And so that's the cartoon that we see here in panel C. This is called the little man, or the homunculus. And this is an illustration you've probably seen before. It's intended to communicate the idea that in the post-central gyri, taking the two hemispheres together, we have a complete representation of the body, but that representation is distorted. Those body parts that provide us with the greatest spatial acuity and the greatest sensitivity to mechanosensory stimuli are hugely represented in the brain, and they're represented in these distinctive features of the post-central gyrus. The hands are represented where the central sulcus has that S shape, where the two gyri interdigitate against each other, and then the face is represented in that inferior segment of the post-central gyrus. Whereas the remaining parts of the body are not nearly as magnified. We call this phenomenon cortical magnification, which has to do with how much more does cortical circuitry magnify the representation of the body that's otherwise related to the density of peripheral receptors and the density of neurons at all the antecedent stations in the somatic sensory pathway. Well, the upshot here is that the homunculus makes very clear that the important somatic sensory surfaces for us as humans would be our hands and our face, especially the facial structures around the mouth and the tips of our fingers. Now, before we leave the primary somatic sensory cortex, I do just want to comment on the functions of these four parallel body maps. You may find that curious, and frankly, a lot of us find it somewhat curious, but there do seem to be distinctive receptive field properties that are represented in these four different maps of the body. One might suggest that area 3, being subdivided into 3A and 3B, is truly the primary somatic sensory cortex from a physiological perspective, whereas areas 1 and 2 seem to process a more higher order aspect of somatic sensation. Area 3A is especially concerned with our proprioceptors, those receptors that are found in deeper structures, such as our muscle spindles and our Golgi tendon organs and our joint receptors, whereas area 3B is more concerned with cutaneous receptors. So area 3B is more cutaneous, is going to be more concerned with light touch, for example, than the movements of our muscles and joints. Now, once we get into areas 1 and 2, we're encountering more complex aspects of somatic sensory processing. Area 1, for example, is going to respond to complex stimuli activating multiple skin surfaces, for example, over multiple fingertips, and perhaps responding selectively to a particular pattern of motion, our fingers passing over a counter surface from left to right, for example. Area 2 seems to be concerned more so with the shapes of objects that we encounter. So it suggests that there's some kind of computation of lower level signals into more higher order signals that begin to build up a sense of structure in our somatic sensory interactions with the world. Okay, and so finally let's consider what happens to this information beyond the primary somatic sensory cortex. What we discover is that the primary somatic sensory cortex, being at this level here, has extensive interconnections with other parts of the brain. There is something that we recognize as a secondary somatic sensory cortex that is in the inferior and posterior aspect of the parietal lobe, perhaps also involving the posterior aspect of the insula. And this secondary somatic sensory cortex receives input from our primary areas. And from there, that information is further elaborated in a higher order sort of way and passed into structures in the medial part of the temporal lobe that are going to be important for making memories and for interacting with our emotional networks. So that involves the hippocampus and the amygdala, respectively. Areas 1 and 2 likewise project into our secondary somatic sensory cortex, but area 2 in particular, the one concerned with more complex geometrical aspects of somatic sensation, projects into parts of the parietal lobe that sit just posterior to it in the superior parietal lobule. And these parts of the parietal cortex seem to be especially concerned with our orientation relative to the environment. So there's a spatial mapping that's going on here in this part of the brain, and that mapping is used to drive our motor system to interact with the world in appropriate ways. So for example, imagining a visually guided reach. That is a means of directing our somatic sensory surfaces towards a particular goal. Well, that's going to be integrated through parietal and frontal networks, most likely involving this superior parietal lobule region. Okay, well, this has been a very quick sense of cortical processing in the somatic sensory system. There are so many big questions that we've yet to address, such as how does perception actually work? How is it that we can have, for example, the integrated sensation that one might experience when holding a snowball or an ice cube, or maybe immersing a finger in a cup of tea? Consider those experiences. We're combining a sensation of shape with structure, with a sense of texture, and a sense of temperature, and just maybe a sense of pain as well. I've been emphasizing the concept of labeled lines and parallel processing, but at some point in our perception, we unite these labeled lines into a solitary percept, and it's still not clear to neuroscientists how that unity of representation occurs. There may not be an anatomical explanation for that perceptual sense of unity. There may be other physiological means to build up that percept, and this is all under active investigation. So hopefully this tutorial has given you a framework for understanding how mechanosensation works, and will also help you to put the anatomical pieces together when we look in a different tutorial in detail at the organization of the somatic sensory pathways. So I'll see you then when we consider that interesting anatomy that gets us from skin to cortex. Well, finally, before I leave you from this tutorial, I want to suggest a study question that begins to apply this knowledge in the clinical domain. I want you to consider what would happen if a patient were to suffer a stroke involving the middle cerebral artery. And I want you to think about such a stroke occurring in the right hemisphere. Okay, now I'm giving you a bunch of options to think through, and they all begin with this word anesthesia. And anesthesia implies lack of somatic sensory sensation, particularly in the domain of mechanosensation. So consider these options and go ahead and indicate in one form or another what you consider to be the best choice among this set of options. I'll see you next time. Welcome back to our discussion of the mechanosensory systems, which are part of our broader systems we have within our bodies for processing somatic sensory information. All of this pertains to several foundational concepts in the field of neuroscience. Two in particular that I would highlight are, of course, our first one that should be very familiar to you now, that the brain is the body's most complex organ, and also that there are genetically determined circuits within the brain that are truly the foundation for the operations of the nervous system. And so we're going to focus in this tutorial on one such circuit. We'll call it a pathway. It's a pathway that allows mechanosensory signals to get from our surfaces where they are detected, such as our skin surfaces on the tips of our fingers, all the way up to the appropriate processing stations in the cerebral cortex. So our learning objectives are for you to characterize and recognize. And you'll see these pairs of terms played out across several distinct mechanosensory pathways. First, I'd like for you to be able to characterize a pathway known as the dorsal column medial lemniscal system. I'd like for you to be able to recognize components of this pathway when you actually confront them in images of the central nervous system, be they views of the brain from the outside, histological sections, through the brain or the spinal cord, or some other conceptualization of elements of these pathways. So here we're focusing on the dorsal column medial lemniscal system, which is a long-term but very descriptive. It tells you about one particular pathway. Another pathway is the corresponding mechanosensory pathway for the face, and this runs through the trigeminal nerve and components of the trigeminal nuclear complex of the brainstem. So again, I want you to be able to characterize the pathway and recognize the elements when we actually look inside the nervous system. And then lastly, I want to introduce you to a pathway that gets mechanosensory feedback about the movements of the body into the cerebellum. The cerebellum is a marvelous organ that facilitates agility, be it agility of bodily movement or agility of thought. And we'll spend an entire set of tutorials thinking together about the cerebellum. For this tutorial, I want to introduce the pathway that gets information about movement from the spinal cord into the cerebellum. And as usual, I want you to be able to recognize components of that pathway when you're confronted with them. And the recognition part is really key in this tutorial. It will be key for the rest of the course, because I want you to begin to build up a mental model of the nervous system. And being able to recognize the components as they actually appear will be key for the success of your mental framework or your mental model. So that way, you can consider what happens when elements of that model become damaged or dysfunctional. And that will help you translate your foundational knowledge in medical neuroscience to the care and treatment of individuals with neurological injury and disease. Well, let me introduce again the pathways, and then we'll look at them one at a time. So for our conscious awareness of mechanosensory stimuli, there are two pairs of pathways. One for the body below the back of the head, including the back of the head. We call this the postcranial body, post meaning posterior to the cranium. And there is a pathway that we call the dorsal column medial lemniscal system. It has elements of the dorsal columns of the spinal cord and the medial lemniscus, an important pathway in the brainstem. Now there's also a pathway that mediates pain and temperature that we'll look at in a separate tutorial for the postcranial body. That pathway is called the anterolateral system. I include it here for completeness. And now for the face, we have a corresponding pair of pathways. For mechanical sensation, we have a pathway that runs through a division of the trigeminal complex of the brainstem called the chief or principal sensory nucleus of the trigeminal complex. So that will be a pathway for mechanosensation. For pain and temperature, there's a pathway of the trigeminal nerve that runs through a different part of the trigeminal brainstem, a subdivision of that complex called the spinal trigeminal nucleus. Okay, well, let's begin by thinking together about this pathway for mechanical stimulation for the postcranial body called the dorsal column medial lemniscal system. Now, we have a view of the entire pathway taken from our companion textbook, and this is probably really small on whatever size screen you're using to view this right now. So let me just give you the broad overview of this pathway, and then we'll look at more magnified views of this figure so that you can see things more clearly. Well, I'll make a few points in general terms. This pathway for mechanosensation involves 3 neurons as we go from skin to cortex. Okay, so I want you to focus on these 3 neurons. I want you to consider where are their cell bodies. I want you to consider the distribution of their axons. And thirdly, I want you to be able to discuss and to illustrate the location of the synaptic connections from one axon to the dendrites of another neuron in the serial pathway. Okay, so three things to focus on in this pathway and all the others that we'll look at over the next few tutorials. That is the location of cell bodies, the distribution of the axons of those cell bodies, and then the locations of the synaptic junctions. So let me just ask you, where would we find cell bodies? Hopefully your answer is, well, somewhere in gray matter within the central nervous system, or perhaps in a ganglion if we're talking about something outside of the central nervous system. That's great if that was your answer. I might ask you, where would we find axons? Well, hopefully you would say in the central nervous system we'd find axons in white matter. And for the peripheral nervous system we'd find axons in nerves. Also a great answer. Well, what about synapses? Where would we find synapses? Well, maybe just a little bit more thought about that leads you to answer, well, gray matter. Synapses are the output zone of one neuron, but it's also the input zone of another. So we would find synapses in gray matter. Great. Well, let's keep those thoughts in mind as we look at this pathway in some detail. So this pathway begins with a first-order neuron, which is a dorsal root ganglion neuron. Okay, so here it is right there in the cervical enlargement of the spinal cord, and here's the dorsal root ganglion and the lumbar enlargement of the spinal cord. These are the first-order neurons. So they have a peripheral process, as we saw in the last tutorial, specialized to establish some labeled line, some particular aspect of mechanosensation. And then the central process enters the spinal cord. That central process enters the dorsal column and ascends. It runs the longitudinal length of the spinal cord until we hit a set of nuclei on top of the dorsal column, and those nuclei exist in the caudal part of the medulla, specifically in the dorsal part of the tegmentum. We call them dorsal column nuclei. Dorsal column nuclei, or DCN for short. That's what we have at this level. So that's where we find the synapses for the next neuron in the pathway. So those neurons then grow axons that do something really important. They cross the midline. And as they do, they establish the location of the decussation of this pathway. So for every pathway we consider, we want to remember that for the cerebral hemispheres, there's a principle of contralateral representation that we must solve. So in the case of the mechanosensory pathway, contralateral representation is established in the caudal medulla with the outgrowth of the second-order axon. It sweeps across the tegmentum of the medulla and ascends the rest of the way through the brainstem and synapses on the next neuron in the pathway, which is in the thalamus, specifically the ventral posterior lateral nucleus of the thalamus. So that's what we have here, our third neuron. So the dorsal column nuclei was our second-order neuron, and our third-order neuron is in the ventral posterior lateral nucleus. So that's our third-order neuron, and from there, the thalamic cells provide input on up to the postcentral gyrus. Once we reach the postcentral gyrus, we basically abandon trying to order the neurons because we recognize that when the thalamus projects to the cortex, we're providing input to a vast network involving hundreds of millions of neurons that potentially could be modulated by this sensory stream. So at that point, given that scale, it just doesn't serve us well to continue the numbering scheme that has served us well along the pathway from skin up through the thalamus. All right, well, let's have a bit of a closer look, and we'll focus in on the first order neuron making a connection to the second order neuron. So again, our first order neuron is a dorsal root ganglion cell. Now we can see a little bit more clearly, I trust, an important distinction between the afferents that arise in the cervical enlargement and those that arise in the lumbar enlargement. The first order axons from the lower part of the spinal cord enter the dorsal column near the more medial aspect of the dorsal column on one side of the spinal cord. As we contribute axons from increasingly superior directions, those axons add to the lateral aspect of that dorsal column. Consequently, when we reach the cervical cord, the input, which is significant, of course, because we're talking about mechanosensory signals from the upper extremity, they simply add to the lateral side of the dorsal column. So there's a medial to lateral progression as axons contribute. As a result of this progression, in the cervical spinal cord, we recognize a division of the dorsal column into two parts. There is a medial gracile tract and a lateral cuneate tract. We'll see them shortly in Sylvius. These two pathways run the length of the spinal cord. The gracile tract runs the entire length. The cuneate tract begins to form in the upper thoracic cord and then runs on up through the entire cervical enlargement. And these tracts meet a nucleus at the top of the column. There's one nucleus on top of each tract. There's a nucleus called the gracile nucleus that sits on top of the gracile tract, and then a nucleus called the cuneate nucleus that sits on top of the cuneate tract. Okay? So that's where we find our first synapse in this pathway, and the second order neuron then, which is part of this dorsal column nuclear complex, grows an axon that sweeps across the midline. And as these axons sweep across the midline, I'll just draw a bunch of them here, we give that a particular name. We call that the internal arcuate fiber system, or the internal arcuate fibers. It's simply the second order axons as they cross the midline. Now once they cross the midline, then they make a sharp turn and project in the superior direction. It's really the very same axons, okay? We just give them different terms in different locations. When they're crossing the midline, they're the internal arcuate fibers. After they've crossed the midline, they form a distinctive medial ribbon of white matter that we call the medial lemniscus, or ML for short. Well, let's see the medial lemniscus as we blow up the top half of this figure from our book. So now we have a indication of at least two axons that are ascending from the dorsal column nuclei to the ventral posterior lateral nucleus of the thalamus, the VPL, and it's this fiber bundle that we call the medial lemniscus. Okay, so again, these are second order axons. They make synaptic connections in the thalamus and the VPL in particular, and then from there the axons project into the postcentral gyrus. And they do so in somatotopic order. For example, the fiber that is projecting here into the paracentral lobule is a fiber that was activated by the gracile tract, and that fiber is communicating somatic sensory signals from the lower extremity. The thalamic neuron from the VPL that terminates here near the middle of the postcentral gyrus is being driven by an axon that entered the spinal cord in the cuneate tract. So this is an input that's being driven by what we did with our contralateral arm or hand. Okay? So the exquisite body map that we talked about in the previous tutorial is established by the systematic ordering of axons from the thalamus projecting into the postcentral gyrus. Okay, well, I hope that was clear. I took some time to go through this quite slowly. We won't take quite so much time as we talk through the next two pathways. But let's turn now from the postcranial body to the face, the anterior 2/3 or so of the head, and ask, what's the pathway that serves mechanosensation for the face? Well, that pathway is called the trigeminal lemniscus pathway, or sometimes we call it the pathway of the chief or principal nucleus of the trigeminal complex. And I'll explain why we call it the trigeminal lemniscus in just a moment. So again, it's a 3-neuron pathway from skin to cortex. Okay, note that conservation of the basic organization of the pathway. So the first order neuron is a ganglion cell, only now it's in the ganglion of the trigeminal nerve. It enters the brain stem through the trigeminal nerve, which, as you should recall, plunges right through the transverse fibers on the ventrolateral surface of the pons. At that level, we find our second-order neuron, which resides in a nucleus of the trigeminal complex called the principal nucleus, or the chief sensory nucleus. The second-order neuron grows an axon that crosses the midline and ascends the remaining divisions of the brainstem, the upper pons and the midbrain, and enters the ventral posterior complex of the thalamus. This pathway we call the trigeminal lemniscus, and it essentially forms near the medial edge of the medial lemniscus, and I'll show you that in just a little bit. Okay, so the third order neuron then is in a division of the somatic sensory thalamus that we call the ventral posterior medial nucleus, or VPM. So VPL was for the medial lemniscus, VPM is the target of the trigeminal lemniscus. You should know where these neurons in the ventral posterior medial nucleus project. They project to the postcentral gyrus, but they project to the inferior one-third of that postcentral gyrus. And as they do, they provide a beautiful somatotopic map of the face across this inferior segment of the postcentral gyrus. So the somatotopy that we see at the level of the cortex really reflects the way the VPL and the VPM map into that cortical domain. Okay, well, let's turn our attention now from our system that makes us aware of our light touch sensations and our vibration sensations and our position sensations to the kind of sensory feedback derived from the movements of our musculoskeletal units that serve to coordinate the ongoing control of movement. And what I'm referring to now is the role of this important part of the brain that we'll talk about in a couple of weeks, the cerebellum. The cerebellum I like to think of as an organ of agility. It helps to facilitate agile movements of body and agile movements of thought. With respect to the body, the cerebellum gets inputs from the cortex about what we are trying to do, and the cerebellum gets feedback from our sensory systems about what we are actually doing. And if there's a problem, as there quite often is, then the cerebellum will generate an error signal that is sent back into our motor system for an adjustment so that some correction can be made as we execute a voluntary movement. So we think of this as real-time, ongoing coordination of voluntary movement. Now, what I want to address is the means by which this cerebellum gets information about how we are actually moving. One important source will be our mechanosensory systems that supply the spinal cord. So there is a pathway that runs up the spinal cord connecting to the brainstem via one of the three peduncles, one of the three stalks of white matter attachment. And what I'm referring to specifically is the inferior cerebellar peduncle. So let's look at this pathway. We consider this pathway part of our spinal cerebellar systems. So these are spinal cerebellar pathways. So the receptor here is going to be a proprioceptor. So it's a muscle spindle afferent, a Golgi tendon organ, a joint receptor, one of those receptors that's stimulated when we move our musculoskeletal units. The first order neuron is a dorsal root ganglion cell, and its central axon enters the spinal cord and, as we'll see in a few weeks, gives rise to some local collaterals in the ventral horn that are important for things like the myotactic reflex or the knee-jerk reflex. But what I want to focus on here is the central projection. So if we're talking about an afferent from the lower body, then the first order axon enters the dorsal column and ascends to the thoracic spinal cord. There is a synaptic connection on a nucleus of the intermediate gray matter of the thoracic spinal cord called Clark's nucleus. It's also called the dorsal nucleus of Clark, so either term is fine. Clark's nucleus, dorsal nucleus of Clark. Okay, I think I want to make an interjection here, and it's based on an experience with the first run we had through medical neuroscience, where some students were concerned about my declaration that the dorsal nucleus of Clark is a nucleus of the thoracic spinal cord, and yet we're looking at a slide that shows a cross-section through the lumbar spinal cord with Clark's nucleus. Now I know that's a small point, but perhaps it's worth clarifying. Clark's nucleus extends throughout the thoracic spinal cord and into the upper lumbar levels of the spinal cord. So this figure actually is perhaps not ideal, but it's also not misleading. There is a caudal extension of Clark's nucleus in the lumbar cord, so it's perfectly fine that we use the lumbar cord for illustration. But I do want you to associate Clark's nucleus primarily with the thoracic cord, and let's just not be concerned with the inferior limit, whether it's L2, L3, or somewhere in that region. I'd be more pleased that you look for Clark's nucleus in the thoracic cord, and let's just leave it at that. And otherwise, let's continue with this tutorial. From there, Clark's nucleus gives rise to a second order projection that runs in the dorsal lateral white matter of the spinal cord. This is called the dorsal spinal cerebellar tract. And this pathway runs right on into the cerebellum via the inferior cerebellar peduncle. So this is a pathway that serves the lower extremity. Now you may be wondering about the upper extremity. Well, the upper extremity is served by a pathway that runs in parallel. So again, first order neurons are dorsal root ganglion cells, and the axons enter through the dorsal root entry zone, but rather than reaching Clark's nucleus in the thoracic cord, these axons ascend in the dorsal column. And from there, they interact with a nucleus that is external to the dorsal column nuclei we've already discussed. It's called the external cuneate nucleus. External cuneate nucleus. Not shown here in this illustration, but it's shown in the next illustration. And the external cuneate nucleus then is the second order neuron that projects to the cerebellum information about the upper extremity. Okay, so Clark's nucleus is concerned with the lower extremity. External cuneate nucleus is concerned with the upper extremity. Now, here's an illustration from my friend and colleague here at Duke, Dr. Nell Kant, where she has tried to put this pathway, the spinal cerebellar pathway, together with the dorsal column medial lemniscal system. And what we see is that proprioceptive information derived from things like muscle spindles and Golgi tendon organs enters the spinal cord via projections that run through the dorsal nucleus of Clark for the lower extremity, or the external cuneate nucleus for the upper extremity. And then from there, the pathways enter the cerebellum via the inferior cerebellar peduncle, ICP for short. Now, as we move our bodies in space, we do have some conscious awareness of those movements. In fact, the movements of our body in space are part of what constitutes our body schema or our body image. So that implies that there must be a way for this information to also enter conscious experience. And there's some concern as to exactly how this happens in the human nervous system, so we don't have a definitive picture of this quite yet. But I think a provisional answer can be that as these pathways, the dorsal spinal cerebellar pathway and the pathway from the external cuneate nucleus, as they arise to enter the cerebellum, they give off collaterals. And those collaterals seem to synapse upon neurons that are either in our dorsal column nuclei or very near them. And from there, the neurons around the region of the dorsal column nuclei grow axons that contribute to the medial lemniscal pathway. And this allows these proprioceptive signals derived from the spinal cerebellar relays to also enter our pathway for conscious awareness. Okay, well, we will say much more about the spinal cerebellar pathways when we talk about the cerebellum in a few weeks. Now what I'd like to do is transition to Sylvius and show you what these pathways actually look like as we ascend from the spinal cord all the way up through the brainstem. Well, to get started in Sylvius, I'm going to open the brainstem cross-sectional atlas and select all structures. And let's begin at the lumbar level of the spinal cord. So I've just selected the lumbar cord, and what I'd like to highlight for you is the dorsal column. So here we have the dorsal column, and as I select it, the label that appears is the gracile tract. No cuneate tract, right? Because we are in the lumbar cord, and we are below the level of the input from the arms, obviously. So the only axons we find here in the dorsal column are those that are serving mechanosensory signals from the lower body, the lower extremities primarily. So if we now proceed to section higher up in the spinal cord, you'll notice that we are adding axons to the peripheral sides of this dorsal column region. So there are some unlabeled axons that are out here laterally. So that's where we're going to find the cuneate tract, right? So the gracile tract, medial for the lower extremity, the cuneate tract, lateral for the upper extremity. So these are elements of our dorsal column medial lemniscal system, conveying mechanosensory signals from the spinal cord all the way on up to the caudal medulla. Now before we get there, let's go back down to the lumbar cord and let's consider the spinal cerebellar pathways. So there may be afferents from muscle spindles and Golgi tendon organs and the like, or deep proprioceptors entering this dorsal root entry zone and entering the dorsal column. But as we ascend in the dorsal column system to the thoracic cord, some of these afferents are going to dive into the intermediate gray matter in a region that is on the medial side of this gray matter. And right about in this area, we have what we call Clark's nucleus, or the dorsal nucleus of Clark. This is the origin of the axons that then sweep out and run from thoracic cord into the cerebellum in a pathway that we call the dorsal spinal cerebellar tract. So there's the dorsal spinal cerebellar tract. Now an important point that I want to emphasize. The dorsal spinal cerebellar tract is on the same side of the spinal cord as the entering afferent fiber. So this allows us to establish an ipsilateral input to the cerebellum from the body concerning proprioceptive signals. Now, we've been emphasizing that in the cerebral hemisphere, the principle is contralateral representation. But I warned you that when it comes to the cerebellum, we have a different principle. It's ipsilateral representation. And this is how it's established via an ipsilateral dorsal spinal cerebellar tract that's relaying information derived from the same side of the body. In this case, the lower part of the body. Now, what about the proprioceptive signals for the cerebellum from the upper part of the body? Well, here we are in the cervical cord. Those signals are going to enter through the dorsal root entry zone and enter the dorsal column in the region of the cuneate fasciculus. And from there, these signals are going to ascend. Let me just select the cuneate tract so you can see what will happen there. They will ascend into the caudal medulla. So here we are in the region of the caudal medulla, and we are now very close to where we would find our full complement of dorsal column nuclei, including this far lateral external cuneate nucleus. So here's our external cuneate nucleus, and I'll highlight it here on the right side of this image to be consistent with where we started. So this external cuneate nucleus that's in view here received input from the ipsilateral afferents concerning proprioceptions of the upper extremity. From here, this external cuneate nucleus is going to grow axons that will join up with those that are running right next to it from the dorsal spinal cerebellar tract. And eventually, we can follow that tract all the way into the cerebellum. And at some point, this tract begins to accumulate axons from the brainstem that are serving other purposes, and for that reason we no longer call it simply the dorsal spinal cerebellar tract, but rather we now begin to call it the inferior cerebellar peduncle. And that peduncle makes its way into the cerebellum, which is where we are now. Okay, now let's return to the dorsal columns of the spinal cord and consider again what happens in the dorsal column medial lemniscal pathway. Okay, so we sort of jumped ahead a little bit to the spinocerebellar pathway. Let's get back to the dorsal column medial lemniscal pathway. So here we have a tracts, the cuneate tract and the gracile tract, that are going to ascend in the dorsal column region of the spinal cord until they hit the caudal medulla. And what will happen is that a nucleus will be encountered essentially on top of this column of white matter. There's a gracile nucleus sitting on top of the gracile tract and a cuneate nucleus sitting on top of the cuneate tract. And if we look a few millimeters above, now we can see that the nucleus is engulfed by the surrounding axons of the tract. So this is where we would find our second order neurons of our dorsal column medial lemniscal system. Okay, so we have tract and nucleus, tract and nucleus. Now, these second order neurons grow out axons that are going to cross the midline and form a critical pathway of the brainstem called the medial lemniscus. Now, as these axons grow out, we call them internal arcuate fibers, but once they cross the midline, they begin to coalesce into a medial ribbon of white matter, and that's what medial lemniscus means, medial ribbon. So now we can follow this medial lemniscus all the way up the brainstem. Here we are through the medulla, and at this level the medial lemniscus is in the anterior and posterior axis of the brainstem. But as we rise through the pons, the anterior part of the medial lemniscus begins to move out laterally. It's as if this medial ribbon is beginning to twist. And as it twists, while we rise superiorly through the pons and enter the midbrain, this pathway is now getting in a position to hit the ventral posterior complex of the thalamus that we'll find in the next section in our atlas. So here's our ventral posterior lateral nucleus of the thalamus, and it's in a position ready to receive the input from the medial lemniscus that we saw just in the previous section. Okay, and from the ventral posterior lateral nucleus of the thalamus grows the axons that enter the internal capsule and then make their way to the postcentral gyrus, allowing for the third order neuron then to relay its signals to the level of cerebral processing in the cortex. Okay, now let's complete our journey through the brainstem for mechanosensory processing by thinking about the signals derived from the face. And for that, we need to get into the pons at the level of the trigeminal nerve. So here are the nerve roots of the trigeminal nerve. So the mechanosensory signals of the first order axons enter the brainstem through the nerve root where they encounter a nucleus. So here's the chief sensory nucleus of the trigeminal complex. And from here, there is a outgrowth of a second order axon that sweeps across the midline and ends up being very close to the medial edge of the medial lemniscus. So somewhere around in here is where we expect to find the fibers of the trigeminal lemniscus. So I'm just going to select the medial lemniscus as a placeholder so that you know that there are some afferents concerning mechanosensation in the face near the medial edge of that pathway. And then as we ascend through the brainstem on our way to the thalamus, that medial edge of the medial lemniscus is now in a position to meet the medial division of the ventral posterior complex. We call that the ventral posterior medial nucleus. So here's our home for our third order neuron. This is where its cell body resides, and it grows axons that join the internal capsule and project to the inferior third of the postcentral gyrus. All right, now you may be wondering, how can I possibly learn all of this information and keep this straight? Well, there is really one key strategy that I would suggest, and that is to draw. I don't care how you draw. You can draw in the dirt if you like, but I would encourage you to draw and to make your drawings as large as possible. The bigger the better when it comes to making visible your knowledge. And as you draw, you can hold yourself accountable to what you actually see from your own hand. So what you produce is what you can then interrogate. And here's how you might go about interrogating your drawings. Let's imagine that you select the left medial lemniscus. Okay, so I'm placing my cursor here on the left medial lemniscus. You might ask yourself questions like this. Where are the cell bodies that grew these axons? Or you might ask, where are the synapses of these axons? Now, I'm going to encourage you to hold yourself accountable to the precision of your language. It's my conviction and my experience that precise language is a reflection of precise thinking, and that's what I want for you, precise thinking. Okay, in answer to the first question, where are the cell bodies that grew these axons? The best answer would be in the contralateral dorsal column nuclei. If we're talking about axons that are in the dorsal part of the medial lemniscus, we might say, well, those axons were grown by the contralateral cuneate nucleus. So if this is the left medial lemniscus, the axons in this position were grown by the right cuneate nucleus. If we're talking about the more anterior or ventral part of the medial lemniscus, those axons were grown by the contralateral gracile nucleus. Or for this left medial lemniscus, I'm referring to the right gracile nucleus. So yes, indeed, there is somatotopy in the medial lemniscus, with the upper extremity represented more to the dorsal and eventually the medial aspect of the medial ribbon of white matter, and then the lower extremity represented on the more anterior or ventral side, and eventually the lateral side as we end up ascending through the brainstem. So out lateral is where we would find the axons grown from the contralateral gracile nucleus, and more medial, the contralateral cuneate nucleus. And then right along the medial edge here at this point, we would find the axons grown by the contralateral, or in this case the right-sided, principal or chief sensory nucleus of the trigeminal. Okay, I hope that all makes good sense to you. Now, what about our synapse question? Where are the locations of the synapses of this left medial lemniscus? Well, the synapses would be in the ipsilateral ventral posterior lateral nucleus. Okay, why did I say ipsilateral? Because my reference here was the left medial lemniscus. I'm asking, where does the left medial lemniscus terminate? The left medial lemniscus terminates in the left ventral posterior lateral nucleus of the thalamus. And one way to speak of that would be to say ipsilateral VPL. Now, some of you may have thought, okay, where does the medial lemniscus terminate? You might have thought, all right, it terminates in the ipsilateral postcentral gyrus. Well, if you're thinking about where does the information ultimately go, it goes to the left postcentral gyrus. But that wasn't the question. I didn't ask you where the information went. I asked you, where do these axons terminate? So please keep in mind that I'm referring to the anatomy here. And the anatomy has a clear endpoint. In this case, these axons physically terminate in the thalamus. They do not project through the internal capsule into the postcentral gyrus. That's the job of the third order neuron, the thalamic neuron. Okay, I hope I'm clear about that. I know some of you will struggle to catch on to this, but the best way to grasp these concepts is to rehearse them, to practice them. So please make drawings however you can. Please explain them to your friends and family. I would love to see you do that. In fact, what I'd like to do now is pause and show you one such example that is out there on a YouTube home. that does just that, explains the dorsal column medial lemniscus to friends and family, and now to all of you. So let's take just a minute and view this.
Hi, I'm Christy. This is my extra credit project for Neuro, OT 441 with Professor Stein. Something you should know about me is that I'm not musical, I don't write songs, and I'm not a poet. But I do want extra credit, so I'll give it a try. I've composed 2 songs about the different tracks that we learned about, and they are both set to children's tunes, so hopefully they will be familiar to you. This first one is about the DCML tract, and to orient you, let me show you that this is a finger, this is the dull end of a safety pin, and this says touch. So we'll be discussing the touch of the dull end of a safety pin on the finger, and the magical journey that it takes, sending the information all the way up to the postcentral gyrus. So let's get started with the DCML tract, which is set to the tune of none other than the Itsy Bitsy Spider. Alright. The DCML tract can be confusing. The UEs are lateral fasciculus cuneatus. The LEs are more medial fasciculus gracilis. And the medial lemniscus is a tract of white matter. First order neuron goes in the dorsal root. Ipsilaterally projects up the dorsal column. Then it terminates in the medulla, where it lands in the nucleus matching the dorsal column. Second order neuron starts in the medulla. Decussates and ascends via the medial lemniscus, up through the cerebrum into the thalamus, where the VPL of the thalamus serves as its last stop. Third order neuron starts in the VPL, through the internal capsule, which is just white matter. Then out it comes and synapses on the brain, on the postcentral gyrus of the parietal lobe. Primary somatosensory cortex, that is where this info finally ends up, responding to touch, vibes, and proprioception. The mechanoreceptors got the job done. DCML.
Well, I think that's fantastic. I'm sure many of you can do the same and even better. But I do thank our friend for being bold enough to put this video and make this available for all of us to see. Welcome back to Medical Neuroscience. Today we're going to talk about the pain systems. This topic is one of the most complex that we can conceive in the whole field of medical neuroscience, so it's not surprising that we're going to be once again reinforcing our core principle that the brain is the body's most complex organ. We're going to be talking about the circuits that arise in early brain development that give rise to our perceptions of pain, and then what we do with those perceptions as pain becomes part of what our cognitive faculty now integrate into our integrated perception of self. And of course, the human brain endows us with a natural curiosity to want to know how all of this works. And as we begin to discover more about the nature of pain processing and the interactions between our pain systems and other aspects of cognition, we're on the threshold, I believe, to really understanding how to best manage individuals who suffer from pain of various sorts. So it's a very exciting time to be learning about neuroscience and its applications in the world of healthcare. Our learning objectives today are to be able to discuss with some foundation the complex phenomenology of pain. I want you to specifically be able to describe the 2 basic categories of pain that we often differentiate clinically. They're called first and second pain, and explain the neural foundation of each. I want you to be able to characterize the peripheral and the central mechanisms underlying hyperalgesia. I want you to be able to characterize the neural mechanisms for the feedback modulation of nociceptive processing. And I also want you to consider the feed-forward modulation of pain processing. And lastly, I want you to be able to discuss the affective dimensions of pain and identify the neural systems that are involved in pain affect. It might be useful in our discussions to consider the nociceptive aspects of pain from the affective or the emotional aspects of pain, and it's from the latter that we believe arise the notion of suffering. So let's begin by discussing the complex phenomenology of pain. Well, we're going to spend a little bit of time on the neurophysiology of the pain systems, and we will begin by considering pain as one submodality of somatic sensation. And the word we use to describe this is called nociception, which means pain perception. But we know that the phenomenology of pain goes well beyond nociception. There is an affective component to pain that has bearing on our emotional lives. And then there's a phenomenon that we call secondary affect, which refers to how the impact of chronic pain begins to work its way into our emotional life, especially as we consider the future consequences of living in pain. And all of this, of course, happens within a psychosocial context that can have a significant impact in modulating the experience of pain. So I think you get the idea that to understand pain, one needs to understand the transduction of signals in our nociceptive pathways, but one needs to go well beyond the concept of nociception. Well, for those of you that are headed towards careers in the healthcare systems, there is quite a lot of change that has taken place over the last decade and will likely to continue to take place as the impact of pain in healthcare management continues to evolve. We've seen a radical change in the way that we assess pain, at least here in the United States. Thankfully, assessments of pain is now part of standard procedure every time a patient enters the healthcare system. But even the best practice for how we assess pain is not so clear. There are changing strategies for pain management, and I think we're seeing a welcomed introduction of interdisciplinary approaches in pain management. And I think that's going to be a key strategy towards dealing with not just nociception, but the consequences of pain as well. And as a result of all this, I think we will continue to see a changing role for healthcare professionals in the management of pain. Well, I share this with you just to give you the broader context of the problem of pain in medical neuroscience. Okay, well, let's begin to talk about the neurophysiology of pain, beginning with the neurophysiology of nociception. Well, as you remember from last time, nociception begins at the peripheral ending of a dorsal root ganglion neuron. And in the case of our pain and temperature afferent fiber, that dorsal root ganglion neuron grows an axon that ends in a structure called a free nerve ending. The central process extends into the spinal cord where there is a synaptic connection with a neuron in the dorsal horn. And note, that's an important distinction between our mechanosensory pathways and our pain and temperature pathways. The mechanosensory pathway extends a central process that enters the dorsal column and then grows up to the caudal level of the medulla before making a synaptic connection along the mechanosensory pathway. But for pain and temperature, the first synapse is right here in the dorsal horn, and we will come back to that point. For now, let's turn our attention to this receptor ending. And what we discover is that within the tissues of the body, there is a type of ending that we call a free nerve ending, because these free nerve endings are not encapsulated, they're not associated with any particular specializations of Schwann cell lamellae, nor are they affiliated with special cells, such as Merkel disc cells. So they're free nerve endings. They distribute fairly broadly within the tissues, suggesting that they have a fairly large receptive field size relative to some of our finer processes like these Meissner's corpuscles, for example. So these free nerve endings, these are the structures that will transduce the signals that lead to the generation of action potentials in our pain and temperature fibers. Well, let's look at those fibers. The pain and temperature fibers are among the smallest sensory fibers that we have in our spinal and cranial nerves. They come basically in two classes, one of which can be further subdivided. Those classes are the A delta fibers and the C fibers. The A delta fibers include axons that end in receptors that are sensitive to mechanical stimulation that can lead to nociception. There's also a type of A delta fiber that is sensitive to both mechanical energy and thermal energy when it threatens to damage tissue. Now notice that these A delta fibers are a little bit thicker. They have an axon diameter of a couple of microns. They tend to be lightly myelinated. In contrast, our C fibers, on the other hand, are very small indeed, and most of these are very poorly myelinated or perhaps unmyelinated altogether. So that suggests that the conduction velocity of the A delta is going to be greater than the conduction velocity of the C fibers, and indeed that's the case. We'll come back to that point about speed in just a moment. So these C fibers, they are polymodal. And what we mean by that is that they respond to multiple modalities of energy or of chemical conditions that potentially can signal damage to the tissues of the body. So they respond to thermal, mechanical, and chemical stimuli. So let's think now about the transduction mechanisms at the ends of these axons. So what we find at the ends of these axons is a type of channel that was discovered not too long ago, and it's a type of channel that is a member of the transient receptor potential family of ion channels. So we call these trip channels for short, and there's a whole variety of trip channels that have since been characterized. These trip channels might be sensitive to one of a variety of stimuli. They may be sensitive to heat. There are trip channels that are sensitive to cold. There are trip channels that are sensitive to the presence of protons, indicating acidic conditions developing, often secondary to tissue damage and inflammation. And then there are modulatory sites on these channels that can bind organic compounds such as capsaicin. So capsaicin is a substance in chili peppers that give it its heat. And isn't it interesting that we've come to use this word to describe the experience of eating a chili pepper? We say that it's hot. We say that my mouth is burning, when in actuality this substance is modulating the activity of a trip channel that otherwise might very well be sensitive to heat. So it seems like we've gained some insight into neuroscience through the culinary arts. So I think that's a wonderful idea. Okay, so these trip channels then can be modulated by a variety of substances, and they can be gated via the interaction of these channel proteins with protons, heat, cold, other factors that are released by the mediators of inflammation in damaged tissue. And what these channels do when they are open is they allow for the passage of cations like sodium and calcium. So as sodium enters the cell, it will depolarize the cell, produce a generator potential and potentially an action potential if threshold is crossed. Calcium, as you will recall, is an important mediator of second messenger signaling within the cell. So as calcium levels change within the terminals of these free nerve endings, then there may be a variety of consequences with respect to protein phosphorylation and dephosphorylation. And that will be important for understanding the impact of inflammation on the sensitivity of these free nerve endings. Now let's move to consider what happens in the central nervous system when the central process of these first-order axons enters the spinal cord. So here we're looking at a cross-section through the lumbar level of the spinal cord, and we imagine that somewhere out here is a dorsal root ganglion cell that ends in a free nerve ending, and the central process enters this dorsal root entry zone and synapses on neurons in the dorsal horn. And I'll just highlight one very interesting part of the dorsal horn. I've told you elsewhere that the dorsal horn is a series of layers of cells, and one layer that's very important in processing signals related to pain is called layer 2 or lamina 2, and that's identified on the left side of this panel. This is called the substantia gelatinosa, for the appearance of this layer in a myelin stain, such as what we're seeing here. So it appears rather bright, meaning that it's densely populated by small cell bodies, with the exclusion of most heavily myelinated axons. So within this region, this lamina 2, we have a rich assortment of projection neurons and interneurons that are going to be receiving the input from these first-order axons. This circuitry of the dorsal horn is going to be an important mediator of signals coming both down from the brain as well as feeding forward into the spinal cord that will modulate the transmission of pain signals from the first-order axon to the second-order cell body. So we'll come back and talk more about that circuitry near the end of this tutorial. Here's yet a more schematic look at this circuitry here in the dorsal horn. So there are our pain and temperature afferents, our C fibers or A delta fibers coming in to the dorsal horn, and here's our substantia gelatinosa, our layer 2, and deeper down we have additional cells that receive this incoming information about pain and temperature near the base of the dorsal horn in laminas 5 and 6. And notice that the C fibers, they tend to terminate more superficially, mainly in what's called the marginal zone and in the substantia gelatinosa, this more posterior cap of the dorsal horn, whereas the A delta fibers tend to terminate more deeply on these large projection neurons that are found in lamina 5 and 6. The A delta fibers might also give rise to superficial collateral. So I think it's helpful to just remember that the C fibers predominantly terminate out in the region of the substantia gelatinosa, whereas the A delta fibers tend to project more deeply at the base of the dorsal horn. Well, from both places, from the more superficial parts of the dorsal horn as well as the deeper part, second-order axons arise from neurons that will then project all the way up through the brainstem and terminate among targets there and in the thalamus, as we'll see in more detail in a different tutorial. But for now, what I want to do is highlight the difference, we think, from a functional and clinical perspective between the signals conveyed via the A delta fibers and those conveyed via the C fibers. Now, imagine the unpleasant scenario of banging your thumb with a hammer, or perhaps closing a door on your fingers. That's going to hurt. And the way it's going to hurt is that there will typically be a very rapid, sharp shooting pain. So we call that first pain. And that first pain typically subsides in just a matter of seconds. And what's left is more of a throbbing pain that we call second pain. So there's this first pain and a second pain. And we think that these two qualities of pain that follow slightly different temporal dynamics correspond to signals that are transduced by A delta fibers and C fibers. We think the A delta fibers are associated with the first pain, and the C fibers are associated with transducing the second pain. We know that because in experiments when it's possible to silence or to take out the A delta fibers, all that's left is the second pain. And in complementary experiments where the C fiber has been silenced, what we have is the sharp shooting pain, but not so much this dull aching pain that evolves more slowly and persists for a longer period of time. Well, as it turns out, the dorsal horn neurons that are receiving this input from the A delta and the C fibers seem to give rise to different connections. The dorsal horn neurons that sit down in those deeper layers, layers 5 and 6 of the dorsal horn, give rise to projections that travel directly to the thalamus. And from there is a relay in the ventral posterior complex, sending signals into the somatic sensory cortex in the postcentral gyrus. So this is a relatively rapid pathway for the transmission of signals from the A delta fiber to the thalamus and then on to the somatic sensory cortex, where the localization of injury can be quite precise, because this information is now being elaborated within the context of this exquisite map of the contralateral body surface that's found in the postcentral gyrus. Now, in contrast, the C fiber system gives rise to a much more diffuse set of projections, and we might have inferred that that would be so if second pain is associated with more of an emotional or affective component. One might imagine that the C fiber information should gain access to those parts of the brain that are concerned with emotion, and indeed they do. The C fiber pathway enters the dorsal horn synapses in the more superficial parts, like the marginal zone and the substantia gelatinosa. And from those more superficial layers, projection neurons grow axons through the spinal cord into the brainstem, and along the way, these axons terminate among a variety of structures in the brainstem, structures that have widespread influence over the activity of cells in the forebrain. Some of these fibers will also make it to the thalamus, but rather than synapsing in the ventral posterior complex, they tend to synapse in medial nuclei in the thalamus, those that likewise give rise to widespread projections to parts of the brain that are more concerned with aspects of cognition than they are with the localization of somatic sensory stimuli. Those regions are in the ventral and medial parts of the forebrain, what we might call the limbic forebrain. So these pathways then suggest something about the functions of our A delta and C fibers. They seem to provide information that runs in parallel but serves quite different purposes, with the first pain pathway helping us to localize the source of the injury to our tissues, whereas the C fiber pathway seems to motivate a response to that injury. So now let's have a different schematic look at these pathways. So on the left side of this figure, we have an illustration of the first pain pathway. So again, this begins with an A delta fiber that is going to provide input into the dorsal horn of the spinal cord, and then from there will be a direct projection from spinal cord to thalamus. This side of our anterolateral system is called the spinal thalamic tract, because it's a direct shot between the dorsal horn of the spinal cord and the principal thalamic nuclei that then relays signals up into our somatic sensory cortex. Now, as you might imagine, things are a little bit more complex on the other side of this figure. The other side of this figure is intended to illustrate the second pain pathway. So here's our second pain pathway, which begins with C fibers that provide input into dorsal horn neurons that then give rise to projections that terminate in a variety of structures throughout the brainstem, and even on up into the forebrain, into structures such as the amygdala and the hypothalamus. Now, we won't talk in depth about all of these targets, but I do want to highlight two in particular. The reticular formation refers to a broadly distributed set of cells in the core of the brainstem, and among those cells are multiple small nuclei that are involved in very particular kinds of functions. And some of these functions are those that influence our overall level of arousal and our overall level of attention. And it's through such mechanisms, we think, that the reticular formation can receive information about pain and thereby modulate our cognitive states. There's another interesting structure in the brainstem that receives this information. It's called the periaqueductal gray. The periaqueductal gray is a structure in the midbrain that surrounds the cerebral aqueduct. We'll come back and talk more about the periaqueductal gray in a few minutes. This is a structure that's critical for the top-down or the feedback modulation of pain transmission in the dorsal horn of the spinal cord. Now, over here on the right side of this bifurcating input is nuclei of the thalamus that is on the medial side of that Y-shaped lamina, in particular nuclei right along the midline, and nuclei actually buried within that lamina. These nuclei give rise to cortical projections that are accessing parts of the brain that are involved in processing emotional signals, as well as signals that help build up an image of our body that is especially informed by our internal state. That seems to be what's going on here in the insula. Whereas the anterior cingulate cortex is a very interesting part of the brain. It participates in prefrontal cortical networks that are involved in evaluating the significance or the consequence of our actions. And we know from some very interesting studies that activation of the anterior cingulate cortex seems to be associated with the burden of our pain. And this can be modulated in experimental studies through hypnotic suggestion. And it's been demonstrated that the more we feel pain as an unpleasant experience, the greater is the activity in this part of the cingulate gyrus. Okay, well, I just want to review for you again the overall organization of somatic sensory pathways, and I'm going to refer you to a separate tutorial to go into more detail about the organization of the pain pathways. But I'll just remind you that for the conscious awareness of somatic sensation, we have two pairs of pathway. One pair of pathways that serves the postcranial body, and another pair that serves the region of the anterior cranium, including the face. So for the postcranial body then, we have a pathway that is concerned with somatic sensory experience in the domain of mechanosensation. And this would be the dorsal column medial lemniscal system that we discussed in a previous tutorial. Our system that conveys pain and temperature signals for the postcranial body is called the anterolateral system. And now for the anterior cranium, for the region of the face, our mechanosensory pathway runs through the principal or the chief sensory nucleus of the trigeminal complex, as we saw in a previous tutorial. But for pain and temperature signals, the processing runs through a different division of the trigeminal brainstem. It runs through the spinal nucleus of the trigeminal complex. So at this point, I would encourage you to just hold the thought about the pain pathways, and we'll take that up in a separate tutorial. But for now, let's move on and consider some topics of particular interest concerning how nociception is processed and modulated by both local factors as well as neural factors. So let's consider the phenomenon of hyperalgesia. Hyperalgesia refers to inflammatory pain. This is a condition of enhanced sensitivity to mechanosensory stimulation that follows the onset of injury and the development of a response in the tissue that engages the immune system and other mediators of inflammation. So hyperalgesia is due to the peripheral sensitization of our nociceptors, largely due to the action of paracrine mediators of inflammation. So following tissue injury, there's an immune response to that injury. And there are immune cells that will infiltrate the area, such as our macrophages and our mast cells and our neutrophils, and they begin releasing immune mediators that can have an impact on the vasculature that supplies this region and have impacts directly on the free nerve endings of our first order nociceptive axons. Many of these mediators are those that will modulate the activity of our TRP channels, things like ATP, bradykinins, protons, histamine, prostaglandins. All of these mediators can increase the currents that flow through those TRP channels and thereby increase the sensitivity of those free nerve endings. Well, notice what else is happening here. The free nerve ending itself can release neuroactive peptides that then interact with these immune cells or the vasculature. So there is, in a sense, a bit of bidirectional signaling here between the inflamed tissue and the free nerve ending itself. These interactions can lead to an increase in the sensitivity of that free nerve ending to the signals that are being transduced into pain. Well, in addition to peripheral sources of modification that can lead to hyperalgesia, there can also be changes within the central nervous system itself, specifically in the dorsal horn or in the spinal trigeminal nucleus, that can lead to central sensitization. So this can contribute to the phenomenon of hyperalgesia. So there may be activity-dependent increases in the excitability of these second-order neurons following high levels of nociceptive activity. And this increase in the excitability of these second-order neurons might generalize from nociceptive inputs to the collaterals of other polymodal inputs that might respond to mechanical stimulation. And this can be a problem. This can lead to a phenomenon called allodynia, which means that a normally innocuous stimulus might be perceived as being painful. And perhaps this is happening because these second-order neurons now have generalized their excitable state. Now, there seems to be two forms of central sensitization that reflect the mechanisms of long-term potentiation. So you'll recall from our discussion of synaptic plasticity that with a coordination of activity across the synaptic connection, that connection can strengthen. There is an early form of long-term potentiation that's transcription independent. So this is what we consider to be a phenomenon that's called wind-up in the field of pain management. So this is when there's repeated activation of nociceptors and it leads to the sustained depolarization of that second-order neuron. If there is an NMDA receptor-dependent mechanism at play, you can imagine that sustained depolarization might be sufficient to remove magnesium block, allowing for the influx of calcium and the establishment of long-term potentiation at such synaptic connections. So we know that the postsynaptic conductances that are gated by glutamate can become more effective now in these neurons that have undergone these sorts of wind-up phenomena. And so presumably that reflects an activity-dependent plasticity at the connection between these first-order nociceptive afferents and their targets in the dorsal horn. There's also a transcription-dependent form of central sensitization that we imagine might be triggered through that influx of calcium. And that requires the mediation of transcription factors that can modify the expression of genes in the nucleus of cells there in the dorsal horn. So changes in gene expression can lead to a longer-term maintenance of that hyperexcitable state. So now let's turn our attention to the central regulation of nociception and pain. And let's consider the phenomenon of analgesia, which refers to the absence of pain despite the presence of a nociceptive stimulus. Well, as I mentioned at the onset, pain is a complex phenomenon, and it's subject to all kinds of modulatory influences. For example, context is very important. The emotional context of the trauma, such as stress during injury, can lead to a heightened sense of the experience of pain associated with that injury. Your cognitive understanding can have a profound impact on your experience of pain. Perhaps many of you have been injured during some athletic competition, and perhaps you were aware of the injury at the time, but you quickly re-entered the game and continued to participate, and it was only some time later that the full impact of that injury was experienced. So somehow you had the ability to allow context to help you suppress your experience of pain. And then there are cultural differences in the experience of pain. And we know this from studies across the world that have looked at the experience of pain in different people groups, and we know there can be incredible variation in how different cultures establish their norms for the response to pain. We don't know the explanation for most of these phenomena, but at least some of them seem to be attributable to the feedback or the descending modulation of pain transmission. And we are now gaining some insight into the circuitry that's involved. So the feedback pathways that modulate pain are depicted in this figure. And there are a number of structures that are highlighted here, and I'd like to draw your attention to a few of them. One right in the middle is called the periaqueductal gray. I mentioned this briefly. This is the gray matter that surrounds the cerebral aqueduct in the midbrain. The periaqueductal gray is really a key node in a network that allows for the feedback modulation of pain. The periaqueductal gray is receiving input from structures in the forebrain, such as the amygdala and the hypothalamus. These are structures that are involved in our emotional experience and expression. The periaqueductal gray receives input from the somatic sensory cortex and from the insular cortex, and together these inputs are integrated. The output of the periaqueductal gray goes to other parts of the brainstem, in particular the reticular formation that's present in the medulla. And from this part of the reticular formation, we have inputs that terminate in the dorsal horn of the spinal cord. In addition to the medullary reticular formation, there are really interesting inputs that come from some of the nuclei of the brainstem that release biogenic amine neurotransmitters, such as the locus coeruleus and the raphe nucleus. The locus coeruleus releases norepinephrine in the dorsal horn of the spinal cord, and the raphe nuclei release serotonin. And these neuromodulators can influence the excitability of the dorsal horn neurons. Well, this figure is far from complete, and we don't really understand how all of this works, but the basic principle at play here is that there are projections from the brainstem to the dorsal horn that can impact the transmission of nociceptive signals right at the very first synapse in the pain pathway. Also present in that circuitry of the dorsal horn in the region of the substantia gelatinosa are interneurons that release neuropeptides such as enkephalins and other kinds of endogenous opioid substances. What these neuropeptides seem to do is inhibit the activity of the nociceptive afferent as it makes synaptic contact with the dorsal horn neuron. So we can imagine that descending inputs from structures like the raphe nucleus and the locus coeruleus and the medullary reticular formation can drive the activation of these local circuit neurons with the effect of turning off the output from that dorsal horn. And that, we think, is one way that the brain can produce an analgesic effect right at the very first synaptic junction along our nociception pathway. Well, there's a lot of interesting discussion these days in the field of the neuroscience of pain, especially as it relates to various psychological phenomena that have been well studied in other contexts, such as the placebo effect or expectancy effects. It very well may be that these effects are mediated via projections that modulate how this periaqueductal gray then mobilizes the resources of the brainstem to modulate the transmission of nociceptive signals in the dorsal horn of the spinal cord. So stay tuned. I'm sure we will learn much more about these mechanisms in the years to come. And now let's turn our attention to feed-forward modulation of nociception. And this gets us into a really fascinating discussion of an idea that's more than 50 years old now, and it's called the gate theory of pain. And the idea essentially is this, that somewhere in the central nervous system there is competition between the signals that are being driven from our mechanoreceptor afferents and those that are driven from our nociceptive afferents. And this competition potentially can allow for a mechanoreceptor to close the gate on the transmission of pain signals. Well, here's one circuit that was proposed by Melzack and Wall about 60 years ago that attempts to provide a way of understanding why we do something as simple as squeeze a finger after we've hit it with a hammer. Well, the idea is that in addition to the usual projection to the dorsal columns that our mechanoreceptor afferent might provide, perhaps there's a collateral that synapses on one of these small interneurons in the substantia gelatinosa that potentially can inhibit the transmission of pain information from, let's say, an A delta afferent synapsing upon a dorsal horn projection neuron. Well, if we imagine that this A beta fiber conveying mechanosensation fires action potentials that converge upon such an interneuron, then perhaps the effect is to suppress the firing of our C fiber afferent on the dorsal horn neuron. And this conceptually would be a paradigm for closing the gate on pain transmission. So getting back to hitting your thumb with a hammer, our first impulse often is to squeeze that thumb that's just been struck. Well, I think what our impulse is driving us to do is activate mechanoreceptors. And that competition between nociceptors and mechanoreceptors might be playing out in the circuitry of the dorsal horn of the cervical spinal cord as we squeeze our thumb, such that what we actually experience then has more to do with the sensations that are driven by the mechanoreceptors than those that are driven by the nociceptors. Well, this is a rather vague concept that I think still remains to be well established in the neuroanatomy and the neurophysiology of the circuits of the dorsal horn. But the idea itself has been very powerful, and it's led to a number of new interventions that are aimed at closing the gate to pain transmission, perhaps at other levels. For example, one surgical intervention now to address some forms of pain would be to stimulate with a microelectrode the dorsal columns of the spinal cord. So the idea would be that perhaps there is another gate, or maybe a series of gates, at higher levels of processing along our somatic sensory pathways, perhaps in the thalamus, or perhaps in the cortex itself. And by activating mechanosensory fibers, perhaps the gate to the transmission of painful information can be shut. Well, let's move on now and talk about a few remaining topics before we wrap up. And let's just spend a few minutes thinking together about chronic pain. Well, chronic pain is a huge problem in societies around the world. It's the source of untold disability and suffering from people from all walks of life. And chronic pain can usually be conceptualized as either being related to some nociceptive source or not at all. So there is a nociceptive chronic pain that results from ongoing stimulation of nociceptors. You might imagine that there's some ongoing site of inflammation or injury to peripheral structures that continues to drive afferent activity in our nociceptors. Such forms of chronic pain would stimulate both our A delta and our C fibers, and so one would expect both first and second pain sorts of signals. There's also a chronic pain syndrome that has largely remained somewhat of a mystery to medical neuroscience. Typically, this is a syndrome of chronic pain when there is no known nociceptive etiology. Examples might include some individuals who suffer with chronic low back pain, perhaps fibromyalgia. That doesn't mean that there is not a nociceptive source. It's just that to date, none has been discovered when we apply this label of chronic pain syndrome. The actual etiology, therefore, remains poorly understood, but in most cases, we consider it to be not a matter of a psychogenic etiology, but rather just an etiology that remains poorly understood in terms of the fundamental neuroscience that underlies the source of these signals that are interpreted as pain. There have been a variety of recent proposals about where these chronic pain states might come from. One idea that is under investigation is that perhaps there's some dysregulation of those feedback descending systems that run through structures like the periaqueductal gray and ultimately through the brainstem and into the dorsal horn of the spinal cord. Some dysregulation of the brain's intrinsic means of dampening down nociception potentially could lead to sustaining a chronic pain state. But undoubtedly there's more to it than that, and probably a lot more interesting neuroscience involved. Which leads us to our final topic, and that would be neuropathic pain. So neuropathic pain refers to pain that arises because there's abnormal activity in a nociceptive pathway that's unrelated to the presence of a nociceptive stimulus out in the peripheral structures of the body. Well, one can imagine at least two kinds of sources of this kind of abnormal activity. One could be if there's direct compression on a nerve, and that compression can cause there to be a physical opening of ion channels in the membranes in that region of the compression. Should that be the case, one can imagine the development of an ectopic focus for the generation of action potentials that then propagates centrally, and ultimately the central nervous system interprets that as pain. So nerve compression can be a common peripheral cause of neuropathic pain. Now, if that's the case, one might expect there to be other accompanying sensations, and often that is the case. There are paresthesias, which is a term that's used to describe just unusual somatic sensory sensations that don't commonly map on to the usual experience of encountering a mechanical stimulus. Well, this is one common scenario where neuropathic pain can be trouble, but neuropathic pain can also arise through central mechanisms. So among the central causes of neuropathic pain would be the sustained sensitization of neurons in a nociceptive pathway. We've already talked some about this phenomenon of wind-up. One can imagine if neurons in the dorsal horn of the spinal cord, for example, stayed in that state for an extended period of time, then that might lead to the development of a chronic pain condition. Another idea about neuropathic pain is that perhaps it's a reflection of abnormal autonomic nervous system output to visceral structures, including perhaps even the vasculature. So one idea is that maybe the sympathetic outflow to the viscera is dysregulated in some way, and then visceral sensory inputs back into the nervous system are what's driving this interpretation of pain. But I think perhaps the most interesting idea that's emerged in recent years is the notion that neuropathic pain may arise because of plasticity that's become maladaptive for human behavior. And this maladaptive plasticity might be sustaining the chronic pain state. One prime example of such phenomena might be those that are affiliated with phantom limbs. So phantom limb sensations are those that are generated following a nerve injury or limb loss, either through trauma or through surgical intervention. And here before us are illustrations of what some patients have reported to be the sensations associated with body parts that are no longer present. And you can see that these sensations take a variety of forms. Sometimes there's nearly a complete sensation of the missing limb. Sometimes it's only a partial sensation of that missing limb. Sometimes there's this phenomenon of telescoping, such as what we find here, where the more distal part of the missing limb feels like it's more proximal than it actually would be were the limb intact. Well, these sensations, I think, really capture what I started off telling you about, and that is that pain is a tremendously complex phenomenon. It involves more than just nociception. but perhaps even more significantly, it involves how those nociceptive signals become integrated into our conscious awareness. And that phenomenon is one that I think will continue to be a problem in healthcare, but also I think neuroscience offers wonderful opportunity to approach this problem through new multidisciplinary means. One might imagine, for example, if the problem of pain really is one of maladaptive plasticity in the forebrain, then there may be different ways to intervene that don't necessarily work through the peripheral channel that might have triggered the pain in the first place. Perhaps this is why social structures, why physical exercise often can be such a benefit to people that are experiencing pain states. Well, there's much more research that needs to be done in this fascinating field of pain and pain management. And I hope that you all might have an opportunity to contribute to the advancement of this field and the alleviation of human suffering. But in the meantime, let's apply ourselves well to the studies of the mechanisms of nociception and to the organization of the central pathways that convey nociceptive signals into the nervous system and on up to the levels of the cerebral cortex. So if you've not yet looked at the tutorial on the pain pathways, you might want to go ahead and cue that up next. So I'll see you there when we briefly review the pathways for pain transmission in the central nervous system. Welcome to this tutorial on the central pathways for pain. This tutorial, this subject, relates to a number of core concepts in the field of neuroscience. The complexity of the brain is staggering, and we are only now beginning to truly understand how pain is processed in the human brain, and there's so much that we yet do not understand about this topic. But I'll do my best to explore some of the anatomy of pain as best we understand it. What we're going to look at in this tutorial are circuits that are laid down as a consequence of genetic instructions in the developing nervous system. And we know this because there have been reports of some individuals that seem to lack that genetic instruction and consequently don't experience pain, at least not in the way that most of us would. And of course, we wouldn't be here having this conversation if the human brain did not endow us with the capacity to be curious and to ask questions about our lives and about how our brain interacts with this body and the world around us. And lastly, discoveries in the neuroscience of pain, among other issues that's relevant for human health and wellness, will surely lead to new discovery that will promote healthy living and the effective treatment of disease. So, with that context established, I would say our learning objectives are to characterize verbally, pictorially, and to recognize visually. So we're going to characterize the organization of our pain pathways. An important pain pathway is called the anterolateral system, and this allows us to talk about the transmission of pain from a peripheral nerve right on up to the cerebral cortex. I want you to be able to recognize what the components of this pathway actually look like in cross sections through the nervous system. Likewise, I want you to be able to do the same thing for the pathways that serve the face, and these run through the trigeminal nerve and the nuclei of the trigeminal brainstem complex, specifically a nucleus called the spinal trigeminal nucleus. So I want you to be able to characterize with words, with pictures, in various media, what these pathways are and how they're organized, and I want you to be able to recognize them when we actually look at histological sections through the brain, brainstem, spinal cord. Okay, well, I'll just remind you of the broad organization of our somatic sensory pathways. We have two pairs of pathways. One pair serves the postcranial body, one pair serves the anterior cranium, namely the face. So for the postcranial body, below the head, and including the posterior portion of the head, there's the dorsal column medial lemniscal pathway for mechanosensation. And what we're going to talk about in this session is the anterolateral system. This is the pain and temperature pathway for the postcranial body. For the face, the mechanosensory pathway runs through the chief or principal sensory nucleus of the trigeminal complex. The pain and temperature pathway runs through an inferior division of the trigeminal complex called the spinal trigeminal nucleus. So I'm going to want you to get familiar with those distinctions, both in the brainstem and in the spinal cord. Okay, we briefly introduced our central pathways for pain when I talked more generally about our pain systems, and we recognized that there are two qualities of pain, two categories of pain, a first pain and a second pain. That first pain was sharp, it was somatotopically localized, and it was fast. Well, that first pain is associated with activation of our A delta fibers, and those A delta fibers provide input to the dorsal horn of the spinal cord. And from the dorsal horn of the spinal cord, we have neurons that grow axons all the way from the level of the spinal cord to the level of the thalamus. Specifically now for the postcranial body, the ventral posterior complex of the thalamus, the ventral posterior lateral nucleus to be most specifically. So because of the direct connection between spinal cord and thalamus, this pathway is called the spinal thalamic tract. And once these signals are received by the thalamus, there's a synaptic connection, and a third order neuron then relays information up to the somatic sensory cortex. So this is relatively rapid. This is the transmission of this fast, somatotopically precise information about what body part was injured. And the somatotopic precision about this is attributable to the fact that this information ultimately gains access to this exquisite mapping of the contralateral body that we find in our primary somatic sensory cortex in the postcentral gyrus. Well, that's what we call first pain, or this sharp, somatotopically localized, but thankfully usually transient sense of sharp shooting pain. The pain that typically follows is more of a dull and an aching nature, and the fact that it feels so differently and often persists for a longer period of time suggests that perhaps these are signals that are percolating through a more widespread set of brain structures, and indeed that's the case. Our second pain pathways then have access to a much broader array of brainstem structures, mostly associated with the reticular formation of the brainstem, but also a variety of other structures, including this important structure, the periaqueductal gray, that is one place where top-down feedback signals might limit the transmission of nociceptive signals in the spinal cord. Well, these second pain pathways are distributed all the way up into the forebrain. Structures like the amygdala and the hypothalamus can receive information about the presence of this painful stimulus. Once this information gains access to structures like the amygdala and the hypothalamus, then it's in a position to have more widespread impact on the physiology of the body and in our cognitive state. In addition to projections that are entering the brainstem core and the medial part of the forebrain, there are projections that are reaching medial parts of the thalamus, those parts that have projections up to the insular cortex and to the anterior part of the cingulate cortex. This insular cortex is quite a fascinating part of the brain. We'll talk briefly about it when we talk more about the visceral motor system. It seems to come to represent parts of our body, namely our viscera, that are often in mind when we address questions like, how are we feeling? Well, those feelings tend not to come so much from the skin surfaces. They tend to come more from our guts, our inside. And we think that insular region is part of the brain that's integrating these somatic sensations from visceral sources as well as more peripheral sources, and contributing to our emotional sense of well-being that often gets modulated by pain. Well, all of this complexity on the right side of the figure is really meant to illustrate the impact of second pain on our cognitive status and the processing of pain within the context of our emotional life and our thoughts about the future. Well, thankfully, the anatomy on the right-hand side is much less clear from a point-to-point perspective, so I won't attempt to talk any more about that detail in this session. Rather, what I want to do is I want to take us through our first pain pathways, first for the postcranial body, and then for the anterior part of the cranium, for the face. So let's look first at our spinal thalamic tract. So the first order neuron is a dorsal root ganglion cell, and we've already considered in a previous session the distribution of the free nerve ending in the tissues. So we're not going to look at that here. Rather, we're going to focus on the central termination of this fiber in the dorsal horn of the spinal cord. So that's really the first and most important point to make, and that is that the first order afferent of our pain system terminates in the dorsal horn. So that's where we find our first synapse, not up in the caudal medulla, but right at the level of entrance of that first order axon. Actually not illustrated here, that first order axon may bifurcate and travel up and down perhaps as much as a segment or two in the spinal cord, but not much further. Soon after that distribution across a couple of spinal segments, there will be a synapse. And so the second order neuron is going to be within a segment or two of the dorsal root that attaches to the spinal cord and supplies that axon its entrance into the central nervous system. Well, once that second order neuron receives synaptic connection, then the anterolateral system is generated. And so these second order neurons, they grow out an axon that crosses the midline of the spinal cord just anterior to the central canal. We call this region the anterior white commissure or the ventral white commissure of the spinal cord. These axons then continue around the ventral horn of gray matter, and they enter what we call the anterior and lateral white matter of the spinal cord, hence this compound name, the anterolateral system. Well, once these axons enter this anterolateral white matter, they make a sharp turn in the superior direction. And then these axons run up through this region of the spinal cord white matter, all the way up through the brainstem. Here they are, passing through the anterior and lateral tegmentum of the brainstem, and they synapse in the thalamus. Specifically in the ventral posterior lateral nucleus of the thalamus. Now, probably not on the very same neurons that received input from the medial lemniscus. That would not be helpful, would it not? Because then a sense of touch might elicit a sense of pain, should there be some crosstalk there. Well, there may be some crosstalk, and that might be the basis for some of our referred pain patterns. But for the most part, we think that the anterolateral axons are terminating on a separate set of cells than those that receive input from the medial lemniscus. The VPL of the thalamus, then, would be the source of our third order neuron in this pathway. So the third order neuron then sends its axon into the somatotopically appropriate region of the postcentral gyrus. And from there, this information about this sharp shooting pain can be somatotopically localized due to access to this somatotopic map that we find there in the postcentral gyrus. Now let's look at the comparable pathway that handles first pain signals generated in the face. These signals are transmitted along the axons of the trigeminal nerve, and they enter the brainstem through the trigeminal nerve root. But the central process of this trigeminal ganglion cell doesn't synapse right at that level of entrance of the trigeminal root. Rather, it grows an axon in the inferior direction, and this axon makes synaptic contact with a column of cells that lie just on the medial side of these descending afferent fibers. So this descending tract of afferent fibers is called the spinal trigeminal tract, and the column of cells that receive synaptic input is called the spinal nucleus of the trigeminal complex. Now, there is some somatotopic order along the length of this nucleus. So this illustration perhaps is just a little bit misleading. We may not have a single axon that gives off synapses along the entire length, but rather there may be different axons terminating at different superior to inferior levels of the spinal trigeminal nucleus. And along that length would be a map of the surfaces of the face and the oral region. All right, well, once we get into that spinal trigeminal nucleus, then the pathway bears some resemblance to the anterolateral system. There's a cell body at whichever level we happen to be considering that grows an axon across the midline, and that axon enters the lateral tegmentum of the brainstem and ascends. And that axon will continue to grow in the superior direction until it reaches the ventral posterior complex of the thalamus and makes a synaptic connection in the VPM, the ventral posterior medial nucleus. So this is where we would find our third order neurons that then project into the inferior segment of the postcentral gyrus, which is where the face would be represented. So once again, we have access to a somatotopic map so that we can carefully localize the source of the stimulation. Now, if you followed along well with the discussions of the dorsal column medial lemniscal system and now the anterolateral system for the postcranial body, then I hope you can appreciate the consequences of a very particular injury that produces a very characteristic set of neurological signs and symptoms. The injury that I have in mind is damage to one side of the spinal cord. So this illustration here is of the spinal cord, and imagine that there is some kind of lesion in the lower thoracic area that takes out half the cord. And we haven't yet talked about the motor pathways, so let's not consider them at the moment. Let's just focus on our somatic sensory pathways and think about what we're going to see in a patient with a spinal cord injury that is restricted to one half of the cord. This is actually fairly common. We can see this with a contusion injury, with vertebral fracture, with a tumor that may be compressing the spinal cord from one side. So it's a relatively common presentation, and it's very distinctive. So what such a unilateral lesion would do is damage the ipsilateral mechanosensory afferents that are entering the spinal cord and ascending in the dorsal columns. Meanwhile, that unilateral lesion would take out the axons of the anterolateral system that are grown by the contralateral dorsal horn neurons. So as a consequence, a unilateral spinal cord lesion produces something that we call dissociated sensory loss. The dissociation is a dissociation of a deficit in fine touch perception and pain and temperature perception. So the cartoon to the right-hand side is intended to illustrate the deficits in a patient that might have suffered this kind of injury. In the lower half of the same side of the body, we would have reduced mechanosensation in these various domains of mechanosensory stimulation. On the opposite side of the body, below the level of the lesion, we would expect to see reduced sensation for pain and temperature. So that's the dissociation. Loss of pain and temperature on one side, loss of mechanosensation on the other. Now, if there were a focal lesion on one side of the spinal cord, we would anticipate there to be a limited zone of complete sensory loss simply because we may be damaging the dorsal roots directly, or the dorsal column and the dorsal horn together at that same level. Well, when you think about it, and I'll give you plenty of opportunity to think about it, there are very few places in the central nervous system that can produce this kind of dissociated sensory loss. Perhaps there's a limited region in the brainstem where one might imagine dissociated sensory loss for the face. I would invite you to think about that on your own. But for the postcranial body, if you see this pattern of loss of pain and temperature on one side, loss of light touch, vibration, position sense on the other, there's a very high probability that what we're dealing with in such a patient is a spinal cord injury. If you're following along with the table that I gave you in the tutorial notes for this session, you'll notice at the bottom of the table there's discussion of a visceral pain pathway that may come as a bit of a surprise. So I want to conclude this brief session talking about visceral pain. Well, visceral pain is derived from A delta and C fibers that are innervating the viscera, and such fibers exist throughout all of the body's viscera. And the central processes of these axons for the postcranial body, for example, enter the spinal cord, synapse in the dorsal horn, and give rise then to signals that arise in the anterolateral pathway. Well, that's been what had been understood for many, many years about visceral pain. But more recently, some very careful neuroanatomical studies in animal models, and more recently some physiological studies in animal models, and even some studies done in people have shown that there's an additional pathway that runs not in the anterolateral system, but in the dorsal columns. So this is, I know, a bit of a problem for those of us who like to think that the dorsal columns are all about mechanosensation and the anterolateral system is where we find pain fibers. Now we have to admit that there is a small visceral pain component that runs in the dorsal columns. I say small because quantitatively it is a small contribution to the dorsal columns, but it might not be small in terms of how clinicians might take this knowledge and then develop novel intervention to treat people that are dealing with rather intractable pain coming from their viscera. Well, here's what the pathway seems to look like as best we can understand it. The first order axons, dorsal root ganglion cells that send an axon in through the dorsal root entry zone, but they seem to make a synapse with second order neurons that reside right around the central canal region, this intermediate gray area of the spinal cord between the dorsal and the ventral horns. These second order neurons then give rise to an axon that enters the dorsal column and ascends to the dorsal column nuclei. And at that level there's a synaptic connection with dorsal column nuclei cells that then give rise to projections that seem to run along the path of the medial lemniscus. And they terminate in a part of the ventral posterior complex of the thalamus that sends input into the insular cortex. And as we'll see in a later session, the insular cortex serves as a bit of a somatic sensory cortex for the viscera. Well, this pathway, as I mentioned, has already had some clinical impact. There are patients that have intractable pain due to abdominal and pelvic floor cancers. And some of these patients have been treated effectively by making a very small neurosurgical lesion in the dorsal column area with the goal of cutting through these second order nociceptive fibers while preserving as much of the mechanosensory afferents as possible. At least one such patient that has been well publicized now received remarkable relief from his medically intractable cancer pain. And while his life was not extended by this procedure, the quality of his life was greatly improved. Well, we've yet to understand the full significance of this dorsal column visceral pain pathway, but it does raise some interesting questions. One phenomenon that ought to be mentioned at this juncture is the phenomenon of referred pain, and this is the idea that maybe there's some crosstalk somewhere in the pain system between the signals arising from the viscera and those that are arising from more peripheral structures in the body wall or the skin surfaces. Well, that site of crosstalk has traditionally been assumed to be the dorsal horn of the spinal cord, and indeed we believe that that is the case. But recognition of a visceral pain pathway, such as what I've just described, suggests that there may be other sites for crosstalk as well, such as the dorsal column nuclei, or perhaps even the ventral posterior complex of the thalamus itself. Well, should that be the case, then perhaps visceral pain patterns reflects the integration of visceral and somatic sensory nociceptive signals throughout the entire distribution of gray matter that receives this input in parallel. Well, further studies necessary to totally sort all that out, but it is worth just considering how pain is processed in parallel and when there might be opportunities for those parallel pathways to exchange information. And while visceral pain patterns are not really something that we attempt to treat, they're tremendously useful by allowing clinicians to have some insight into what might be the true source of pain. Obviously, if you have a patient that is complaining of left-sided arm and shoulder pain, one might consider the possibility that there may be a cardiac origin to that pain. And likewise, pain along the midline of the chest region associated with the sternum, or potentially encroaching upon the left side of the flank, might be related to some kind of acidic burning of the esophagus. So referred pain can be very useful from a clinical standpoint, and I hope this tutorial on the anatomy of the pain pathways has given you just a little bit more insight as to why these patterns might emerge. Well, thanks for your attention, and as I mentioned at the onset, there's much more that we could study with regard to the pain pathways. We haven't attempted to look in detail at the second pain pathways, but I do want us to now to transition to Sylvius and give you a chance to recognize what those first pain pathways actually look like as we see them in the spinal cord and the brainstem. So let's transition to Sylvius now. Go ahead and open up your version of Sylvius if you have it, or feel free to follow along with me. Welcome back, and we're in Sylvius now, so I'm going to open the brainstem cross-sectional atlas by clicking onto all structures. And let's go ahead and have a look at the spinal cord first, and we'll consider the distribution of anterolateral system fibers for the postcranial body. So here we're looking at a cross-section through the lumbar spinal cord. Our dorsal roots are entering in the upper part of this image, which is dorsal or posterior. So here's our dorsal root entry zone. And we see this really bright region here in the dorsal horn. That's the substantia gelatinosa. So out here in the marginal zone and in the substantia gelatinosa, that's where our C fibers are going to terminate, whereas more in these deeper parts of the dorsal horn, that's where the A delta fibers will terminate. Well, from these parts of the gray matter arise the axons of the projection neurons that grow the anterolateral system fibers. So a neuron here at the base of the dorsal horn, let's say this is the left side of the spinal cord, will grow an axon that will cross in this region of the white matter. This is what we call the anterior or the ventral white commissure of the spinal cord. The axons grow across the midline and then enter the anterior and lateral part of the white matter here, just to the lateral margin of the ventral horn of the spinal cord. So this is now what we call the anterolateral system. So the axons in the anterolateral system over here on the right side of the image, those axons were grown by neurons that sit on the opposite side of the dorsal horn, and vice versa for these axons here. Now, while I have this anterolateral system selected in Sylvius, I'm simply going to now section up through the spinal cord, and you'll see that as we collect up more and more axons, this pathway grows in size. Now we're in the cervical cord, and we can see it occupies quite a bit of the white matter in this anterior and lateral region. As we enter the brainstem, you'll now be comfortable with the idea that additional structures are found that are not present in the spinal cord. Some of them occupy the ventral part of the brainstem, including structures like the inferior olivary nuclei out here. So our anterolateral fibers end up in a more of an intermediate position, but they're still far lateral. So we are now in the caudal medulla. As we ascend through the medulla, these axons sit more or less between the trigeminal region and the inferior olive, and eventually other components that occupy the basal or anterior region of the brainstem. So here's our spinal trigeminal system. We'll get back to that in just a moment. Here again is our inferior olivary nucleus, and right between the two is our anterolateral system. So let's continue to track the progression of these axons in the lateral part of the tegmentum. Now these structures are just to the posterior side of all of these pontine nuclei and pontocerebellar fibers. And finally, as we get into the midbrain, you'll see that these axons are getting into a more posterior position. They're still pretty far out lateral, still in the tegmentum. And where they end up is in a position to supply the ventral posterior complex of the thalamus, which is right about there. So there's our ventral posterior lateral nucleus of the thalamus, receiving input from these ascending axons from contralateral dorsal horn neurons. All right, let's reset and consider the organization of the trigeminal counterpart of this anterolateral pathway. So this begins at the level of the trigeminal nerve root. So here's the trigeminal nerve entering the ventral and lateral part of the mid-pons. You'll remember that the mechanosensory portion of this nerve terminates right here in the chief sensory, or the principal nucleus of the trigeminal complex. But the pain and temperature fibers, they make a sharp downward bend at this level. And so what we find is the development of a compact tract of white matter that is in the lateral part of the tegmentum of the brainstem. This is a tract made by the central processes of our nociceptive afferents that are present in the trigeminal nerve. So we have a tract just on the lateral edge of a nucleus, and that nucleus is the spinal trigeminal nucleus, or the spinal nucleus of the trigeminal complex. So let's follow this tract on down through the caudal part of the pons and now into the medulla. And as we progress into the medulla, we continue to find this well-defined tract and then a nucleus that sits just medial to that tract. Along the way, the axons in the tract are diving out and terminating on neurons that comprise this long column that we call the spinal trigeminal nucleus. Now, one interesting feature of this nucleus is that the closer we get to the junction of the medulla and the spinal cord, the more this nucleus begins to resemble the dorsal horn. So in fact, we get to the caudal medulla, and now we see a component of this spinal trigeminal nucleus that looks like the substantia gelatinosa. In fact, we call this the gelatinosa part of the spinal trigeminal nucleus. And then the more medial part of this nucleus begins to look like the base of the dorsal horn. So sometimes we like to say that the spinal trigeminal nucleus is really an extension of the dorsal horn of the spinal cord. So maybe that's some help to you conceptually to recognize that the organization of the second order projection from the dorsal horn to the thalamus really is quite similar to the projection from the spinal trigeminal nucleus to its destination in the thalamus. So now we see the dorsal horn again, the substantia gelatinosa, the base of the dorsal horn. Okay, well, let's now ascend and watch what happens to this trigeminal system. So here's our spinal trigeminal nucleus, and we have different divisions of it which are labeled differently in Sylvius, but now we've got the most rostral part of this nucleus in view here, and we're about to again enter the level where the trigeminal nerve hits. So now we don't have the first order afferents above this level because those pain and temperature afferents are descending. Rather, what we have in the tegmentum is the second order axons. Now, I haven't indicated the precise location of this so-called trigeminal thalamic tract. Sometimes it's called the ventral trigeminal thalamic tract, but it's essentially very near the location of the anterolateral system axons, probably just near the medial edge of these anterolateral system axons, right around there. We know it's going to be in a position to eventually enter the posterior part of the thalamus and provide input to the ventral posterior medial nucleus, which is what we see here. Okay, one final point to mention before we conclude this tutorial on the anatomy of the pain pathways. I want you to recognize the spatial separation within the nervous system between the pathways for mechanosensation and the pathways for pain and temperature. We talked about dissociated sensory loss with spinal cord injury, which is based in part upon the spatial segregation of those pathways, with an ipsilateral pathway in the spinal cord for mechanosensation and a contralaterally derived pathway for pain and temperature. In the brainstem, we have a different kind of spatial separation. We have pain and temperature systems out here in the lateral tegmentum of the spinal cord, of the medulla, but near the midline of the medulla, we have a mechanosensory pathway, the medial lemniscus. We don't yet have the facial component because we're in the medulla, below the level of the trigeminal nerve. I say all this to make the following point. We will see patterns of strokes in patients that afflict just the medial part of the medulla, sparing the lateral part, or vice versa. We may see stroke patterns that affect the lateral part of the tegmentum, sparing the medial part. And you can imagine what kind of dissociations you might see in those patients. One might imagine encountering a patient, let's say this is the left side of the medulla, where one might find loss of pain and temperature sensation on the right side of the postcranial body, but the left side of the face, with no damage at all to our mechanosensory systems. On the other hand, one might imagine a patient that has damage to the mechanosensory pathway for the postcranial body and no injury to pain and temperature sensibilities anywhere. Well, these kinds of stroke patterns make great sense if you understand the distribution of pathways for the various modalities of somatic sensation. So I hope that gives you some additional motivation to take on this challenge and make clear and make visible your understanding of this complex clinical neuroanatomy.